Usually the దానికి 10 ఉంటుంది. But online so it can act as a field and it can also act as a signaling molecule and protect the cells and kill the cells. In fact, the best example is how your white blood cells kill the bacteria that are coming out. Once it enters the bacteria by endocytosis, there are so many organic generation that happens there and that leads to the killing of the bacteria. There also always plays a major role. Next. So FAO's perspective on nutrition is food. The first difference is source of energy to live and be active. Perpetuating an intense of malnutrition leads to severe consequences on both individuals and nation. Very bad citizens are unhealthy country, so no productivity. There is huge economic losses that can happen because there is no productivity next year. So this is a figure made by SAI and millets can meet most of these requirements. So what they are showing, how the plate should be. Just look at how your plate should be. So vegetables should be around 400 grams, fruits 100 grams, pulses and all 85 grams, nuts and seeds 35 grams, fats 27 grams, and cereals and nutraceuticals 250 grams. This is how your plate should be. This is how your plate should be. Your pizzas, pastas, they don't give you this type of stuff. Okay? So try to have this in mind whenever you take the food. So coming to millets. Millets encompass a diverse group of small grain cereals like sorghum, pearl millet, barnyard millet, and all these things. Once upon a time, we used to have more millets. Rice was there, but those who have to do a lot of physical activity in the fields and all, they used to prefer millets. No doubt. They are high calorie. They are also high calorie foods. But what is the difference between other things you will see because of that? Next. So what are the health benefits of different types of millets if you just see? We have foxtail millet to combat osteoporosis because it is rich in calcium and proteins. Jowar, it reduces risk of cardiovascular and polar. This one because of high fiber content. And barnyard millet, it accelerates hemoglobin production. And pearl millet, rich in vitamin D and also iron. Finger millet, rich in calcium and polyphenols. Little millets helps in diabetes. Kodo millet, proso millet. We have different types of millets which are indigenous to this country. Not only in the African countries, but they are also known here and people used to consume them a lot in previous times. So, coming to the glycemic index of various cereals, rice 66, brown rice 55, whole wheat, so on. So, millets gives you 52.7 average. Millets and diabetes. Millets are highly recommended for diabetes. And try to take millets. The favorable, because of the favorable glycemic index of this, and most of the gluconeogenic amino acids maintain glucose levels. If the glucose levels are going, there is a metabolic process called gluconeogenesis. From non-carbohydrate sources, glucose will be produced. So millets can produce more they have more gluconeogenic amino acids. Millets have low GIR because of high phenolic content lipids which delay the gastric emptying. So how we can reduce the shooting of the glucose levels in body is try to have foods which delay the process of gastric emptying. As a result, that is more there in the stomach. Slowly it keeps going to down the intestine. As a result, it is the small intestine As a result, more enzymes can attack and blood glucose can rise. But one of the coarse morphological anatomical structure is this, which causes gastric contents. Most of them exhibit good inhibition of alpha amylase and alpha, which reduces GI levels. There are two things which we have to see for the food. One is the alpha amylase content and alpha glucosidase is an enzyme which is present on the microvilli of the intestine, which will be cutting the disaccharide units or disaccharide units into glucose units that causes. So some of the diabetic drugs, they are inhibitors of this alpha glucosidase. As a result, what is happening? These disaccharides will be going off like that, but there is a possibility that these disaccharides can be broken by the large intestinal bacteria. They are also slight, this one will be having absorption, but more of minerals and all will be happening in the large intestine, but we can save ourselves from the absorption in the small intestine. And because International Women's Day is coming up, I have added few slides which is pertaining to women. So if girls have PCOD problem, try to prefer millets. PCOD, polycystic ovarian disease, because if the glucose levels are maintained properly, PCOD problem can be reduced. Next. And millets and obesity. So certain things have shown that this can help in controlling the obesity too. Next. Millets as prebiotics. Prebiotic is different from probiotic. Probiotic is a bacteria. Prebiotic is a fibrocirculating. Next. They are rich in prebiotics. Next. So some of the millets can reduce anemia, particularly some of these things, millets like bajra and all, they have very good iron content. They have got very good iron content. So particularly girls of 13 years and above and are pregnant women, they prefer these. Millets and osteoporosis. Calcium is rich in finger millets and pearl millets, and magnesium and phosphorus. Some of them are rich in vitamins and antioxidants. So pregnancy and millet diet, don't think that millets are not suitable to women, they are highly suitable to women. Next. And gestational diabetes, in fact, can be controlled with the help of millets. Gestational diabetes, once it is there, the baby growth hampers and the baby will be born with very low weight. And lactation and millet diet, it gives all sorts of minerals and all sorts of vitamins and essential fatty acids, essential amino acids are so. And millets not only for the girls and reproductive age group women, but for also women of menopause. So these will be a very good alternative. Next. So what are the bioactive components? And I just rushed through the slides because these are no I can take this. So bioactive components and nutrients present, if you just see, we have phytic acid, flavonoids, polyphenols, essential amino acids, minerals, irons, and very good source of dietary fiber. If you just take a millet diet that covers all these things, those people who cannot afford fruits, those people who cannot afford costly vegetables, we can afford millets. Unfortunately, millets, which was once poor man's choice, has become rich man's choice now, and increasing the cost again. They are not affordable to common man. So that is the only pathetic thing that teaches the art. Once upon a time, 30 rupees per kg or 20 rupees per kg. Now it is 60 rupees per kg. But what is happening? It covers all. It is something like a whole meal. 1 millet is something like a whole meal. Okay. So there is a big list of phytochemicals, polyphenols, alkaloids, nitrogen-containing compounds, terpenoids. So in these things we have gallic acid, elagic acid. So many things, polyphenols are there. Millets as prebiotics. I'll discuss next. So, how sorghum is a functional cereal or functional grain. We have different types of sorghum, grain sorghum, forest sorghum, biomass sorghum, sweet sorghum. For India, it is the Indian Institute of Millets Research, which is the authority to release any type of sorghum variety, and we had collaboration with them, and we have received some varieties from them to work on this. So these are all the different varieties that they had. Remember, the antioxidant or the phenolic content or the polyphenolic content depends on the outer coat color. So we have white ones and all these things. People are thinking that yellow chocolate is good for diabetes, but in our studies we have seen that the chocolate color, not chocolate color, creamy color, chalk color sorghum is there, white journal under. That journal, the brand contains more of the inhibitors for alpha glucose, inhibition of alpha glucosidase compared to the pacha journal. I was under the impression the pacha journal is good. Pacha journal is good for phenolic contents, which will delay the process of gastric emptying. So, what are the three factors that will not allow sugars to rise? Is gastric emptying should be, the time should be very high. It should have some inhibitors like alpha amylase inhibitors and alpha glucosidase inhibitors. So this pacha contains the process of gastric emptying. As a result, the glucose levels will not rise so sharp. But this etrozole contains lot of phytoestrogens. So this is the method that we have used: dehulling, grinding, and homogenization. Remember, whenever you take a millet, don't remove the husk, outside husk. The pericard, what you call, that is the bran. That contains all the good components. If you remove that, it is as good or as bad as your normal rice, except for the coarse grain that is there. The rest of the things are not. So this is the procedure that we have followed to extract the bran and the flour content of the grains. And this is how we have calculated the approximate composition of the sorghum bran sample versus the sorghum flour samples. And these are the different types. See the elements that are present. So many elements are present in this. Next. So anthocyanin content. The moment anthocyanin, most of them are antioxidants. Look at the anthocyanin content. And remember, just I want to tell you, emphasize, there is a particular antioxidant called, this is also anthocyanin, 3-deoxyanthocyanin. This 3-deoxyanthocyanin can resist the pH changes and still retains an antioxidant activity. Usually, the juices that we take, their antioxidant activity is not because of the original juice content. They are adding some synthetic antioxidants. But 3-deoxyanthocyanin is the one which can resist the changes in pH and still can exhibit the antioxidant activity. So one of my students has worked, if we can add this 3-deoxyanthocyanin, are we able to retain the thing? She has covered it with the fruit juices, different types of fruit juices, and then saw how the antioxidant profile is. So I have also worked on split sorghum juice given by both IPSAT as well as IIMR. So this is the phytochemical content and the tannin content. Next. So again, the elemental analysis for this split sorghum juice varieties, total phenolic and flavonoid contents, and antioxidant activity. And we did some HBS analysis of all these extracts. So this is the comparative data of alpha glucosidase inhibition in bran and flour samples of different sorghum varieties. So the red one is the bran, yellow one is the flour. Anti-hyperglycemic activity of split sorghum juice extracts. they are revolving around the ROS. Remember, why we did to look up this study is, Rajendra, maybe you can also focus. Thing is, in radiotherapy and chemotherapy, what we are doing, we are giving some medicines or radiation to improve, which improves the ROS and kills the cells. Again, you are giving antioxidants. Pandu are rich in antioxidants. So, one gets confused whether to kill or what to do. So, these cereals, if you just look at normal rice, it is not rich in antioxidants. It is not rich in antioxidants. Millets are rich in antioxidants. Millets are rich in cellulosic compounds. So, when we consume them, because millet is also like a normal rice, right? It is a stable food that we take, like any rice item, right? At least 2 or 3 we eat. It is containing a lot of cellulosic compounds, containing a lot of antioxidants. How the cell behaves, how the cancer cells behave in the presence of these two things. Okay. These things. So, next. So some plant-based this ones for the activity. Next. So antioxidants, they try to neutralize the ROS. Next. Several different types of bacteria antioxidants which try to delay the process of aging. So these are some of the plant-based cellulosic compounds. So what we did is we took some of the this earlier MB231 breast cancer events we took, and then we have seen how the oxidative stress is there in this and in the presence of these cellulosic compounds. CA4 and EK4. This is not published, so I don't emphasize more on this. So we did whether this is happening in the late apoptotic stage or early apoptotic stage and all. So what we studied is the WAD canonical signaling pathway we have studied, and we have studied some of the genes and all. So another one that we did is we designed things. In design, we have made a design and developed a nanofiltration based membrane setup by the sarcom syrup preparation. So that is for better storage and all. This is supported by ACD materials. So I just want to tell you that try to have millets, but they are also high calorie diets. Right from the beginning, try to have them. But as far as calories are concerned, less to other carbohydrates. As a result, better physical activity is required. So it is able to address more problems in an Indian context. Millets can address most of the problems in an Indian context. So try to prefer millets. Thank you. Any general questions also is fine. If you have any general queries also is fine.
Any more questions or not? If you want any very one question. PCOD policy. Actually, that is in the even in the concepts. Recently, I have published one paper. In these millets, what is the main component is trying to reduce the PCOD.
So basically, PCOD by giving not only a balanced diet, but it is able to control the carbohydrate metabolism.
For giving a nice sorghum experiments. Thank you, madam. Please be seated. Now I request Dr. Professor from ICMR National Institute of Nutrition to second the session as the chairperson.
Thank you, madam. After a long time, again I am listening. She covered a lot many things to enlighten people. Then she started the lifestyle disease and touching each one. So hypertension, PCOD, diabetes, obesity and all. Giving the basics, and she explained about her work, how the millets work. Really, how many of you are eating the millets? You don't know. Yeah. The millets is Carterville always he says that you have to keep on eating and all. For that one, it is the seed coat is very we have to soak in the water for 8 hours before we cook it. So that what are the antioxidants, what are the components are there, how it is working, anti-cancer effect, anti-PCOD, probably the PCOD. So these are all the mechanisms, whatever when studied, what the prodigy they made, what the things I made with that, clearly explained everything. I hope you understood. Thank you very much, madam, for all your efforts to educate our students. Okay, thank you very much.
Now it is a time for felicitation to Professor 𝕃𝕀𝕍𝔼 from JNU. I request all the faculty of zoology to join to this for the felicitation program. National Institute of Nutrition. Dr. 𝕃𝕀𝕍𝔼 sir, he is presently working. He is a scientist and head of the department animal facility, the ICMR NIL Hyderabad. Sir, he did MSc post-graduation genetics in Osmania University, PhD in genetics from Osmania University. He did postdoctoral fellow in the 1999 Director of Rice Research, ICAR Hyderabad, India. And in the 2000 INRA in France, and he was postdoctoral fellow in 2003 from University of USA, and in the year 2002 CDFT Hyderabad, India. He was group head molecular biology and immunology 𝕃𝕀𝕍𝔼 India in the year 2005. In the year 2006, he came as scientist to National Institute of Nutrition, Hyderabad. The honors and awards received by Sir are in the year 2005, he was invited participant for ITEA workshop at Bangkok, Thailand. He received 𝕃𝕀𝕍𝔼 Memorial Young Scientist Award in the year 2006. He received ICMR Fellowships for Young Biomedical Scientists in the year 2007. He was visiting scientist, that is the Cancer Institute of New Jersey, USA. Visiting scientist in the Rockefeller University, Howard Hughes Medical Institute, New York, USA. In the year 2014, he was nominated member for PCOS brainstorming meeting held at ICMR in Delhi. He was nominated as CTC SEA MOEF and CC member for 3 years period in the year 2015. He was invited member Ministry of College Technical Evaluation Committee in the year 2021. Sir is a member in various professional bodies too. He is a member of International Society for Genomic and Evolutionary Microbiology, a life member. He is a member of Nutrition Society of India, life member. He is a life member in Indian Pharmacological Society, the 𝕃𝕀𝕍𝔼 MOE, and he is a life member in Laboratory Animal Scientists Association as well. Sir has many publications. He is a co-author of many books. One of the book chapters, 6 books chapters, and research papers 60. He is a reviewer of various reputed journals. 9 students have completed PhD under his supervision. Sir has completed projects funded by DBT, ICMR, NIH, USA, UK, from United Kingdom. His research interests are on the genetic and epigenetic approach, impact of nutrition on non-communicable diseases. With this brief introduction, I warmly welcome Dr. 𝕃𝕀𝕍𝔼 sir, scientist at HOD from Animal Facility, SAI National Institute of Nutrition, from there.
So good afternoon. Say good afternoon. I want to say enthusiasm. It's a more interactive than the lecture. Routine lectures you are listening now. So once in a while we come, Uma madam, myself and some others, they invited. Right or wrong? So the purpose of this meeting is bringing people people here. So behind it, a lot of drama will go on. Selection of the speakers and the finances, how to arrange the things and all. For you people, they do all the things. I know how much madam requested me to come over here. So I am here. Finally, thank you madam. అంటే పట్టు వదలకుండా అసలు రావాలి మీరు అంటే కదా. So today I am here. Shall we start? We light now is the story started. I like what you want. Switch off the lights. Is it okay? Visible? Right. So I know Hindi, Tamil, Kannada, so other languages. Telugu badi తెలుసు చదువుకున్నప్పుడు కానీ సాధించిందంటే ఏం లేదు మేడం వివేకానంద ఒకటి మనకు ప్రోగ్రామ్ చెప్పింది. సో అవే అండ్ అవే కంటిన్యూ రీచ్ ద గోల్. ఎస్ we have to reach it. మీరు మాట్లాడితే నేను ఆపిస్తా. రెడీగా ఏదో ఒకటి జరగాలి ఇక్కడ. either you have to talk or I have to talk. Which one you want? Sure? Right. Thank you madam. She made the way forward. Now the story will go in another way. We can see the lifestyle disease, lifestyle disease. Why it is named as a lifestyle disease? I changed my style. We change really our style? Yes. So if you go and see our elders, they don't have any TV and all. After that the things have come into our mobile. We are bending our head like that. Always one finger is getting the pain. Only one finger. Style change or not? Whatever we are eating in our brain we don't know, but we are eating. Wherever we are walking we don't know, we are walking. We are seeing and we are sleeping. So once we wake up immediately we are seeing that one. Without that we die. Chachukuma? Tell about it. Sure? మరొకసారి చెప్తున్నా సీరియస్ ఆలోచించి చెప్పాలి. We have to tell. Without mobile phone I will die. I will not die. సగం మాట్లాడుతూ చచ్చిపోయేది. I can live comfortable. To be frank, without mobile phone I can live comfortable. I want only the 99% for my phone. Because why should I kill myself? Technology is required. The technology should not kill us. This now whatever the mobile phone, this mobile phone is developed by us. It is my slave. Are in front of the slave you are bending your head. Nee bhagavantudu kaluchukoni ivodeni bhai. Is it right or wrong? Don't bend your head anytime. You have to bend the head only 3 times in the life. One is to touch your parents' feet. తల్లిదండ్రుల కాలు మొదలు ఓపెన్ లేకుంటే తలంచకండి. Second is your teachers who are giving the knowledge for us. That's what now. Guru Brahma, Guru Vishnu, Guru Deva Maheshwara. Third is look into the book. Book చదివేందుకు సరస్వతి దేవి మనని కటాక్షించని చదవండి. అంతేదాకా మొబైల్ కోసం నేను తలంచుకొని దొరుకుతా అంటే it is giving the Lord many comments and all. You decide what you want, then how the story will start. So someone will be here or I have to operate. Is it okay? This is my national staff nutrition where I work. You are all welcome to that one. Your college has to set a mentor or director. We would like to visit. You can visit our institution. There is a nutrition division is there. We will give the nutrition education. So these are lifestyle diseases. Madam already explained you. Many things are there. So the lifestyle is changed. That's what I am getting the disease. Corona has come. It is a virus. Virus is created by someone else. And naturally it is undergoing for a change. Hello here, what do you want? Look at me. Here now the virus has come into the picture because some mutation will come. Never virus is there. Always the influenza virus is everywhere. Andariki unda leda. Now we are all getting the throat infection. That is there. We cannot avoid it, but how to overcome is important for us because it will live with us. When new things will come, then we have to act accordingly. So for that now we are here to do, we are all here, we are all scientific community, we have to develop something. For that the first step is our body. For our body we need the immunity. If you are weak, it will affect you. If you are strong, it will run away. Anything else? Right or wrong? Yes. No sir, where is the time to eat sir? I have to look into the mobile and I don't find the time. That's why no lifestyle changes, the disease will come. Not only the lifestyle disease, also the opportunity infections are there, they will attack on us. Next. That is called a communicable disease. Second is a non-communicable disease. Then we will see what are the characteristics of this one. Complex etiology, understanding is a problem. PCOD madam said, you know clinically to identify, we can see the hirsutism, so pimples on the face and all, whereas the real diagnostics is a big problem still today. So etiology we have to understand how it is coming, what is the problem for it. Diabetes, until unless the sugars will go up, we don't know. Hypertension, until unless we get the neck pain and all, we are not realizing. So that now disease etiology is very much important to understand. Like that, no multiple risk factors are there. Communicable disease. The problem is that. So, this is a mouse, huh? This is not this. This is not. By knowing you should not do the mistake now onwards, you should be alert. No, I am not doing any mistake now onwards. I don't want to add to the list. If I adding me, that is my mistake only. So that now, what is the scenario and all graphs? Madam explained for Indian scenario. This is a world scenario. You can also look into the WHO. So thanks to my students who made all the slides. So like now, what is the world scenario? How it is day to day increasing? Why it is increasing? Some we cannot avoid. We can avoid. Yes, everyone is busy, everyone is having the stress, everyone is having the problems. But even though there is a way around. So this is how the obesity and the all lifestyle diseases are increasing. So that, okay, to take one example of this one, the comorbidities of all these things, not only one. If you become obese, madam told what adipose is doing. What adipose is doing, excess fat is there in the body. Excess number of cells are there. When the more money is there in our market, we have the main ideas to eat the biryani, go to the cinema, or change the mobile phone, or something. When fat is more, the fat what it has to do, it will keep on secreting the unnecessary things, more than whatever we want. What the hormone has to do in the body, it will go and trigger somewhere. So that now the problems will come. Better not to come now. So why you want the fat? Sir, I don't have the time to work. Yeah, we don't have the time. Everyone is having the 24 hours. I am also having the 24 hours. So this is a problem for us. We are not listening to our body. So that's what now these many are there. Not only that, but the brain, heart, the kidney, it is hitting everywhere. So that for this one, Indian obesity is going up. Sir, what can I do, sir? It is available in the market. Madam said pizza, burgers. I said biryani also there with us. Every day eating this biryani is good for us? Not correct, no? So that no diversity is there. That's what in the nutrition lab, we are always educating you people to eat the diverse food. So, when Chinnam there is saying is there, Unayat Pachpur Seta, Nenat Pajareta, Pango Chinat Parmananeta. That is true. We have to see the diversity is there. Because our elders are bound up, ancestors bound up. Money is to roll rate and go away. Always we want the same type of the food, same taste, same masala, same spices. Is it a bad thing? No. That's what now, when you are choosing, it is choosing us. It's so this is happening for that one, no? where the obesity is a major problem, we are increasing the weight that leads to the obesity and all. Is a genetic variation. In this classroom we are not same. So if I eat the same food, I am going to get the same thing. Question will come. No. To satisfy ourselves, we say my body nature is like that, I will be like that only. That is wrong. Not only the body nature, along with whatever we eat, wherever we live, how much we sleep, whatever we drink, everything is matter for us. All the factors are environmental factors are working. We cannot avoid. We can see seed also different colors of seeds are there. One is genetics, second is environment. Those colors we can see environmental is there. If you go under the Himalayas, wherever the low mountain bottom, the color is different. When you take to the upstairs, then the color will change automatically. This is called paramutations. Mutation is a genetics one. There are paramutations are there. So this is easily erasable. Madam told about the pre-diabetic. So pre-diabetic will come to the normalcy. It is not a problem. If you don't follow, will become the diabetic. So that now we can see not only that one, they are also there. Now Varangal is very good in that temperature everything. So soon we are going to get the coach factory in the Caliphate. Yes madam, I want to talk to you. మీ ముందు కూర్చోబెడుతున్నాను, నేను కూడా మాట్లాడతాను. నేను వచ్చింది ఇక్కడికి మీ మేడం పిలిచింది ఎందుకు, నా కళ్ళు చూస్తూనే ఉంటాయి, గుర్తుపెట్టుకోండి. You have to talk to me. Here this is changing. When the industry will come, it will give the smoke and pollution. What the insect has to do? It will change its color for your information. In evolution we studied, it is changing the color. Why it is changing? It wants to survive. But we are not changing. We want the same. That is the problem for us. So that here tulips are very nice. These are all very good. Whatever it is growing in Hyderabad, the same thing if you take the wire and the size will change. The temperature is little more here. Why? Temperature effect, environment effect. What you eat is effect, what you drink is effect, what you smoke is effect. All are there. So we considering all these things, then we see to understand all these things. Innocent fruit fly we kill. That is the Drosophila eyes. Drosophila is having the 21 type of the eyes colors. These are genetically changing. How environment will change, how the genetics is there, both we will see. Why this is happening variation? Why we are getting the variation? What could be the reason? Food. Yes. We cultivated, we ate. Now it is the pack in the bottles. How many of you are having the habit of reading the food labels? Food pocket పైన ఏముందో చదివిరే ఎప్పుడైనా? ఎన్నడైనా చదువుతారా మీరు? చదవరు. చదవకుంటే ఎక్కడ ఎడ్యుకేషన్ రాదు. Everyone, there is a guideline for the FASI. NID is preparing many things for it. So you have to read it before you eat. Whether to eat or not, you have to decide. Then here we can see, we cannot stop it. Now, earlier we were 100% agriculture in this country and 20%, maybe 15 to 20% is a Gurukula version is there. We were having a Gurukula education system, nicely we lived. Invaders came, they destroyed us. They want the money from us. We are rich. Now taking the time they are changing, industrialization is taking place. Now AI has come. Now AI summit is going in the country. Now our Prime Minister is invited many countries. The people are here to invest in our country. Not only the jobs, same time the environment also important for us. So that to changing things we have to welcome. Same time we have to take care of our health. So that now when you are not listening to your mind, your clock will twist. It is a twisting. You are not listening to you. Are you listening to you? నీ మైండ్ చెప్పింది వింటున్నా అదే చెప్పండి. ఎందుకంటే చెప్పండి. These are question comes. So then NIA will do the research, Human will do the research, and you will also do the research. The research will come into the matter now when you are not listening. So that now your proteins will change. So your DNA is changing. Because now you are all changing, your habits are changing, your environment is changing. So we want all these things. I want a nice food, very good food. So if you eat all these things, all our elders and ancestors are very good. There is no problem at all. So now things are changing, sir. How can we change? Sugar, salt, okay, maida and these are all came into the picture. Oil. how much we have to consume? Sir, taste is important for us. Health is important or taste? By knowingly again we are doing mistake. Sir, Mysore Boda is nice, sir. There is only Maida. Maida is difficult. Sir, we want a more oil to put it. Without oil, we will die. Take the nuts. If you take the nuts, oil will come into the body. So that every day morning, if you take 10 grams of the sesame seed, no one will palli will no. Ever don't remember, sir? Oil cover. So that no, these are things, sugar, salt, required. How much? We never follow. That's what we never read. Then my style is changing. Naturally. Yeah. So we want a good processed foods are there. Now that no radio joke is there. No TV was a bigger. That time we were like this. Now TV is a small and smart. We are the boys. So that was lifestyle changing. We cannot change. But the food is there always. All type of food is there. But your choosing is important. No sir, I love to eat the pizza and burger. Eat. Who said no to you? Your wish. I am not saying. See, in the market everything is sell. We cannot stop it. What you want to eat, you decide. So decision making is yours. When we are not listening to that one, the things will come. Sir, now we have to go in the flight, sir. Yeah, I used to walk. Then the horse cart, bullock cart has come, cycle has come and all this. But till every day morning you have to walk, doctors will tell. Well, we also know, if you want to go to the next home of your friend, you take the vehicle. We don't walk. To go to the market, we don't walk. Our psychology, no? Anything else? I am telling you a new thing. There, if you want to go to other city, yes, it is required. I went to America. I cannot go that many 1,000 kilometers. So I have to go by flight. But if I want to go to friend, to be frank, I am telling you, 3 kilometers radius, I never use any vehicle. Sir, you are a scientist. Yes, I am a scientist. I am a busy person. I know how to manage my time very well. So when you are changing your style, the things will change. You are eating, whatever is there, what is happening in your body, you can see. These are all there. So these are all things are changing. Now, transition has come. We cannot avoid it. Can we avoid? Sir, Sadhvi చెప్తున్నా సార్ మొత్తం బొమ్మలు చూపిస్తున్నారు బొమ్మ సదే ఉంటుంది. This is the thing. What is happening that your habits are lot of confusion what to eat, what not to eat. So big question is there. అదే ఏమి సావాలి రావాలి ఎటువంటి సావాలి. What is the problem for us? These are the things are there which is eating our brain. Now I can say again there. ఎక్కడికి పోతే అవన్నీ అక్కడే ఉన్నాయి. Are there or not? మీ కన్ఫ్యూజన్స్ ఆన్సర్ ఉందా లేదా? మరి ఎందుకు తినట్లేదు? ఎందుకు తినట్లేదు? So that now your attitude is changing. So you separate your attitude. Then, sir, big problem, sir. Yes, problem. Because our mind is not listening to us. What to do then? Sir, we studied the food web, sir. Really we studied. 8th, 9th, 10th class we studied or not? What to eat, what not to eat. If you don't listen to NI, we do the research. Definitely do. Not only that, our madam has to teach in the JNTU and she will run to the NI to collaborate with us and work. Double boarding now. Maybe only in NI. I know that because I know madam, that's what I am telling. All not only that, but teachers also will approach us. So for that now, so that you want to see your DNA should be like that. I mean, ma'am, that genetics and DNA, my language is the DNA. So that now, yes sir, these are all the things are there. Where we can see unhealthy weight and all, many risk factors are there. So imbalance is happening, calorie in, calorie out. We are taking the calories going out. So the balanced life. Is it the balance is there? When we are taking the calories, for that we have to work. The physical activity is important. How much we have to eat, we have to decide. When we are not following that, the mismatch will come. When the mismatch will come, so there is an energy imbalance is there, your lifestyle disease will come. So that we will put the question. Yes, red tip. Okay, we can say these are all on that a tip on the iceberg. We can say that how it will melt one day. Now we are looking, okay, I have not problem every day I'm eating whatever I love to eat. Okay, it looks like that. When you are growing now, things will totally disturb. So that now it is like ice. So who is there inside? Genetics is there. All may know. No. My body nature is like that. I cannot help it. Yeah, that's true. Your body nature is there. How much is there? So then always being a geneticist we say 1 to 99%. Genetics is there. 1 to. You may have the 30%, 40% all. So that no, another problem also there. Not only the genetics. So there we can see epigenetics is there. Environment also. We have to live together. Environment is what we eat, what we drink, what we sleep. These are all there. They are all together. It is deciding your lifestyle. So for that, environment and genotype is deciding your phenotype. Your phenotype, you are looking like this. I am Rajendra, I am looking like this. Because of the two things are here. My genetics is there, my food habits, my physical activity, my thinking and everything is there. So together it is deciding. For that, can we do some research? A question will come for us. Environment is deciding. For that, then we can see, yes, I can do. My area of research interest is dietary interactions. What it is doing, high calorie is going into my body. What it is doing in the body? Excess calorie is there. Excess calorie leads to many things. Excess energy will produce. Then that leads to the, so ultimately we can say, yeah, more fat is accumulating. So how? So for that we start working on that one. So environmental things we can see. Here 3 rats we can see. One is the whatever natural one, the black one. So like that is brownish and more brown one. Brown one is the obese. B12 deficiency is there in that. Oh, environment is changed. You are choosing what to eat. When you are choosing, you are not taking amount of the B12. What happens? That when you take, it will reverse back. So this is, we have to realize that what we are, environment effect is there. Genetics is there for all, but no deficiency is there. Your genetics is changing, your metabolism is changing. So it is important to understand how the allelic variation is there. Your allele means gene. How gene and environment is working together to decide. Always we, so break our brain. A lot of data will come. We keep on doing the research. Madam showed a lot of data. So she made my work easy. I did not bring that much of the data. So we get a lot of data. In that we have to keep on looking into that where the changes come, where the changes come. So for the change, we try to understand the change. For that, the question we put is dietary interactions we are trying to understand in our lab. So that we have taken the 3 types of rats. Genetically, these 3 are different, not same. The other 3 are different rats. If these rats, if I give the same type of the diet is same. I am giving the same diet. In this classroom, you are all here. If I give the same diet to all of you, same food I will give. Some people will become obese, some people will have the normal. Why? So for that we have started looking into that, how much energy they are taking. So how much food efficiency ratio, how much they are utilizing it. Efficiency to utilize. So on that basis, then we can see body weight varies. In the case of the Bristol rats, we can see one is the control, HF is the high fat, HS is the high sugars. Third is the HFS is the both, mix of both. Calories are same. So that in that we can see 3 types of rats are there. WNM rat, Bristol rat, Fisher rat and SREE rat. There we can see Fisher rat is not having much effect. Whereas we can Bristol rat is having the effect. Why? The body nature of that one is, yes, it prone to develop the obesity in that. So weight gain is more, adiposity is increased, fat levels are increased in that. So that no, for that we are trying to understand the mechanism. We did the histopathology for it. So we have taken the liver fat and whatever the fat it will come, The fat, you know that only abdominal fat is there. We have the different type of the fat. Under the skin also the fat is there. We take that. Gonadal fat we take. So these are all type of the fat we take, and what is happening in the fat? What it is secreting and all, we move into all the things. So that here we can see how the inflammation is changed. So your inflammation is changing because of the high fat? Yes, definitely. What it has to do in the body? It will be hitting you. So what time we are developing the lifestyle disease? The problem is coming. So sir, no sir, I am eating, yes eat. Then I have to go for the physical activity, do it. Then balance it is not a problem. When we are not following, the problems will come. So then we do this type of the research to understand what is happening in that. So plasma pro-inflammatory cytokines, IL-6, TNF-alpha, IL-beta, these are all found to be significantly increased high in the WNN. Only in one rat. Why? The genetics is known to that one where it is developing that. So IL-10 and all, these are the anti-inflammatory. No, when the problem is coming, our body is having some defense system. Some extent it can defend. All you know the type 1 diabetes, genetic defect. We have to take the insulin. Type 2 diabetes is a lifestyle disease. Your style is changing. When your style is changing, then ultimately what is happening? There is a mismatch between the sugar and insulin. So when sugar and insulin is mismatched, then you are developing the diabetes. So how to overcome that one? Physical activity is the best thing. Are we really doing? So for that we can see how the madam explained very clearly the mechanism of the always. We saw that no always what is happening in the T-bath and all. So we can see that no WNN is more having the, so here the antioxidant properties are there. Here oxidation stress is there in that more in the WNN, because when it is eating the calories, more calories, it is not able to tolerate it. So that it is becoming obese. There not only about the genes also we studied in that level beta HSD gene. So the gene levels are elevated. So that no, these are all prone to development of the obesity in these rats. So then fasting glucose levels, fasting insulin levels are changed. So there is a discrimination, there is a change in this one because of the high calorie environments. So for that, so ivermectin and leptin, ivermectin is having the protective effect, but is that also changing. Leptin will there always not. If you are eating more, you say no, don't eat, then your brain will give signal. Morning first lecture they say no, neuro, what is neurotransmitters, then he explained about that. Here now it is explaining you, no don't eat, but are we listening? I love to eat the bhaji, eat more. Why you want to eat more? So when you are eating more, the problem comes. Our body is giving the signals. What is happening to those things also we studied in that one. So then we can see we have given a glucose challenge. Really they are tolerating. WNN is no, not able to tolerate. It leads to the development of the diabetes in that. So this is what we can see how they are all changing the insulin sensitivity levels and all. So how it is regulated. Regulation is there in the class regulation. Silence means silence. It is a regulation by our teacher. In our body some molecules are there. They are regulating our body metabolism. Even though when we don't listen, the problems come to us. So for that, what are the regulatory mechanisms also we studied by different genes and all. I am not going for the mechanisms to understand that how the, so this is the adiponectin, P13K and all different genes we saw in WNN, and opposite one which is not developed the obesity, the Fisher 344. How the triglyceride levels are there. Fat, we can see no cholesterol and all, we see no T levels. What is happening? In which side is more when compared to the Fisher? So these are all leads to development. Not only sir, if I take more calories, if I don't do the physical activity, this many things will happen and yes, it will happen. Yes. That is there in front of you. I published a lot about. Madam has given you answer already. Eat the millets and do the physical activity. Already really? How many of you are finding 5 minutes a day to do your exercise? Being in the used to say everybody. You have to tell the truth. I do. Every day I do. Very good. Remaining, everyone has to make the habit of doing every day 5 to 10 minutes. Generally suggested is 20 minutes. body exercise. Warm up your body. Then we can see again what is happening to the liver tissue. Liver damage is there. We studied that also. There we can see how we can see the fat body is not only in the this one. In the liver also it is going in. More fat bodies are there, no blood flow is not there properly, they will get the cardiac problems. So that no excess fat is not good. So high calorie environment only in the WNN is developing because the side interactions we studied. Not only that, the total genes we see, how many are down regulating and up regulating. The fashion of the molecular biology. Everyone wants to study the molecular biology. We study the genes. How many are up regulating, how many are down regulating. In that, by using the different bioinformatics and all, we have chosen some pathways. We studied the AKT pathway. Then, so we have concluded some of the things. For this one, I am explaining one pathway where we studied high calorie environment. It leads to the stress, oxidative stress, we will see inflammation, altered glucose metabolism, altered lipid metabolism. Then finally, it is altered the mTOR signaling pathway. mTOR signaling pathway generally leads to the cancers they study. But we studied in the obesity. Obesity also leads to the cancer. The triggering point, whatever we identified in our studies. No, sir, fat is having that many things mean this. It is giving many problems for us. So that now, so these are the what we studied. Then, no, sir, I am becoming more fatty. I want to cut the calories. The saying is there, Annamo Ramchandra, Devda Annam Kavalanu. Right or wrong? When we are eating more, the problem we want to cut that. If anything will happen, I want to look smart and slim. But the first thing is, I never think about my exercise physical activity. I want to cut a calorie. Sir, I am eating only half the food. You want a slim or smart or you want a good health? Hey, say it loudly. Sure, huh? Sure. Think once again and tell. You want a slim or smart or good health? All of you want good health? Then listen this story. Then what I did, the first 20 to 30 days we worked on that, how it is developing, why people are cutting as a dish, what to cut, when to cut, protein, carbohydrate, lipid, when morning, evening and all. We are having the wonderful Govardhan Diksha. I am writing a review article now. So my students are working on that. What are the Govardhan Dikshas are there? In our Nirjala Govardhan is there, Ekadasi Govardhan is there, Nomun Govardhan is there, Shivaratri Govardhan is there. We are having all these things. A systematic. But now we want, no sir, I will not eat for 1 week, after that I will eat again. This 15 days I don't want to eat, I want to eat only the fruits. You are deciding yourself without having any basis for it. No, since morning I am fasting, evening I eat the, only I am not eating the rice and I can eat the pizza. These are the things are there. If you cut the calories, what happened? So then what we have taken, the taken is that I want to, so there are many questions will go on. Always I learn from you people to put the questions. Whenever I go to my friend's place, my seniors children are getting married. So when I go, most of them are in, you know that we are all biology people, so we are best when compared to the IT people. They get the job at 22 years. My kids also there. So they will be very happy. Whatever that they are going to the gym or walking and all, they will disappear because they got a job. A small story. So this is I am telling you. After that what happens? When more money is there, more friends will come. So they go to the different places to eat. The first 6 months will go on. They are all disappeared. Minimum exercise, walking and all gone. One vehicle will come. Walking also gone. So calorie is going up. Day to day more calories are there. Unknowingly is becoming overweight. They will join in the job. Join job. They leave the target for them. The target is there by March, February 28. I want the report on my table. They will be at close. You know the IT people? So they have work day and night. Whatever is there, they have done. They keep on eating. Because of free food in IT, most of the IT sectors they give the free food. So when they find little time, they go to the club or something to with the friends and all. So they have the social gatherings. Unknowingly they are becoming obese. So they reach some extent at the time. Now come to my friends kill them. Uncle, tell beta, I want to get married. Good beta, chalo congratulations. No uncle, I am 80 kgs, I want to decrease my weight. Okay beta, decrease. Uncle, I want 60 kgs. Okay, decrease. And 1 week I want to decrease 20 kgs. Is it possible? Tell. Is it possible or not? How can you become? Then I put, Nana is very tough. 1 week is very now I am a nutrition scientist now. So uncle, you take 1 more week extra. They give some concession to me. Is it correct? That was a question started for me. It is eating my brain. So I started discussing with my student to put a question to get the points. So then where we started, when they are decreasing the weight suddenly, they are going for the marriage. What is the fate of the children? I started putting the question. What memory they are giving to the next generation? So that where we started looking into that. So let them cut down. This now to write one project will take much time. So finally I ended now. Then I wrote that concept is the male is a culprit, not the female. Female will not eat because now when the biochemistry is in the pregnancy time they have the nausea problems and all. But the male also a culprit. That's what the question and I put down should be. That's what why I am telling you here how to answer questions. How the question has come to me when I go to my friend's place. When I go to my friend children, thanks to them, they have given the research idea to me. So then I started working on the preconceptional right. So preconceptional right importance, all they say eat properly. So the calorie restriction in a non-pharmacological way they are cutting. In the way we are aging, which slowly we stop eating less, we eat less. That's what the longevity will increase. So when in the case of the preconceptional, where we are cutting down, so that when the female is there, they are not eating, everyone will say eat properly. But the male is cutting the calories. I want to become suddenly lean. I want to marry normal healthy girl. So that healthy girl is there, but what is happening in my body? How my sport morphology is changing? My sport biochemistry is changing. What memory is taking? How my environment is changing? Environmental effects. Genetics is same. Genetics will not change. So that I started looking into that. I put the question. That does it the similar effect of the maternal calorie restriction? So that causes the obesity in their offspring. Does the energy metabolism pathways are affected? This type of questions we put. Then we looked into the things. Not moving. Yeah. Some of them are inside. No, it is a ventilation. Slide. Back in the humble. No. మళ్ళీ మీరు ముఖ్య పెట్టున్నారు లేదు సార్ చాలా స్లైడ్స్ ఉన్నాయి ఎంటీ ఎక్కడ మీకు దీని తర్వాత దాని తర్వాత రండి నెక్స్ట్ రండి రండి దీని తర్వాత స్ట్రైట్ అది కన్వర్ట్ దాని తర్వాత దీని తర్వాత నో ప్రాబ్లం ఓకే అదే Signal lo చెప్తాను, don't worry about it. I love to teach without slides. That is my passion actually. ఎవరు చెప్తారు? మీరే అడిగి క్వశ్చన్స్ సాగలేదు. క్వశ్చన్స్ అన్ని మీ నుంచి రావాలి. The thing is, here what we identified is that now, so when we are cutting the canvas, we studied that in the animal model. First we have given the effect type for the animals. We made it obese. After obesity, when we cut the canvas, how we come for a conclusion? So then I am interacting with the people. Some literature says that I eat 50% only. So 50% whatever we are eating, then we have given the normally how much it will eat, then that 50% we cut. So that now always some rule to follow. Then 50 will become the 60, then 40. Then I put the cut point as a 40, then I give the 20%, 30%, 40% and 50%. Our scientific advisory committee is there, they said no, just look into the 40 and 50. So when I have given this one, when we are cutting the calories and all, so what is happening? The calories we are cutting, whereas it is also losing the micronutrients. Minerals and vitamins enough we are not getting in the food. So that now that will the problem, your metabolism is changing. So that pups got to that one has become obese. They got a thyroid problem at the early ages. So that now, for all of you, listen carefully. Because now, suddenly cutting the calories is not good. You have to decrease your body weight. To grow my body weight, I have taken the 6 to 8 years time. At least I have to take 4 years now. Yes or no? Not a 4 days, not a 40 days. The problems are there. Here we studied the methylation. Environmental effect will give the DNA sequence is same, the tails of the DNA will change. So that is where we studied the DNA methylation changes are changing. So the gene expression levels in the WNM we can see leptin gene we studied. Why leptin gene we studied? It is giving the signal in the brain. So that now when methylation is there, it stops giving the signal. So we keep on eating. We are cutting the calories, we are not feeling hungry, we are cutting the calories. That's the problem for it. So that then we studied here how the energy sensing metabolites are changing in this one. We can see in that obese, normal obese, cutting, lipid beta oxidation is decreased. Lipogenesis is again elevated this one. So in the severe calorie restriction, 50% and 40%, the 40% is going along with the control as well. So that now the 40% means moderate calorie cutting is advisable. Not as you wish. You want to go for more calories, it is not good. So it is worth is so that under the members in this. Then we see the study shows that under the severe calorie body but have gone into the energy saving mode. No. Your body is depleting everything, so that now it has to protect itself. The body is whatever the fat is decreasing. So for that now the protection method, it is giving your fat, it is melting and it's taking the energy from that one. That is not good. Suddenly you are going for downing of that one. So that the energy saving mode might have the imprinted in their genes. Why the DNA has come? Methylation. Because energy is changing, your tails are changing to one. It is making a temporary memory. The temporary memory will go up again if you eat properly. So pre-diabetic madam explained to you. When you go for a walking and all, again you will become normal. You will not become the diabetic. Lifestyle diseases are no change the style, we can come into the normalcy. So then, here elevated fasting, insulin and glucose along with the delayed clearance of the glucose. So these are all we notice in this. What we notice in the obese one, in the calorie restriction also these are the adverse effects. So this is what we publish this, how the SREE pathway is affecting in this. In SREE pathway are important genes which are in the case of the longevity, in the aging, when we are cutting down, systematically these things are going on. But unnecessarily we are going for the calorie restriction, all useful things are disturbed. So that now, when we are cutting the calories, we should be bit careful to go for this, not to improve the inflammation and the lipid oxidation and all will change. So then we can see our studies are demonstrating that WMA is more prone to the obese. Why? Because its genetic nature is that one. According to your body nature, what to eat or not, you have to decide. Not only that, preconception and calorie restriction. In high-fat diet, it is obesity restored as disregulates the metabolism of the offspring via AMP pathway. So we studied the pathway, how it is affecting in that. Here, then what we say, give the message is, what you eat is what you are. You are like this, what you are eating. You agree or not? So whatever you are eating, you are yata anna tata mana. In Rigveda it is very clearly written. So yata anna means the annam, whatever we eat, tata mana. Your manu is there, already is there in our Upanishads, Vedas are there, but we never follow it. So when we are following all this, then but the study adds the genetic makeup of our individuals are important. Now water balance we are telling, no, yes, you are correct, but your genetics also important, environment also important, both are deciding your fate. So for that, what to do? Way forward. The prevention and control and management of the lifestyle disease. Only the prevention is the best method. For that, being a student, if I study every day, being a student, what is my duty? Education or anything else? Say loudly. No. How many of you reading the textbooks? Raise your hand. How many of you reading the books? Sir, being a student, my job is to read the books. When you are not doing here, you can say here management is important. Sir, I have the dream to become the Ramanan is there. Right? So I want to become a scientist. I wish you all have to become more than me. I feel very happy being a teacher. We market Gopal like a happy feel. When I see my teachers at school level, I go and tell them big why? Because of them today I am here. Now the thing is that now the Prime Minister of India is telling, yes, we have to achieve the goal because it's Bharat. Are we in that time? So we are killing our time by looking into the mobile phones and where we are wasting our time. We are not managing the time, we are not eating properly, we are not sleeping properly. When you are not sleeping properly, the stress will come. Stress. Prevention for that one, no? First you sleep 8 hours. Second is physical activity. Third is the diet. I can say diet is lost. So the prevention and management of lifestyle diseases are very forward for the reducing the risk factors associated with the effective health education. Health education. All we know. What to eat, what not to eat, we అందరికీ తెలుసు. తెలుసా తెలియదు? నేను చెప్పండి. తెలిసి తప్పు చేసేవాళ్ళని. This is the what we want to use. Sir is asking. Sir, what to eat? Sir, simple answer is simple. We have to see whatever the seasonal fruits and vegetables. Season is giving the fruits and vegetables for us. Are we eating really? How many of you are having the habit of eating a fruit every day? रोज को फ्रूट देना चाहिए पड़ेगा रोज को फ्रूट एनी फ्रूट ओके so madam showed that NN micrate showed in NN micrate we mentioned that 100 grams sir jina revan jina sir okay good no problem perennial yearly once it will come eat it no problem sir I also give to you 10 kg watermelon is study sir matlab that's what there is a limit for everything 100 grams. So there is a way forward for it. Eat that one. Seasonal fruits are it is giving season for according to us and leafy curries also there. Visit our main website what to eat, what not to eat very clearly we mentioned in that. So well and also physical activity is also important. I told about the stress is another thing. Always we compare with others. It is good or bad you decide. My life is my life. Your life is your life. There is no comparison in life. I am telling you, we are 5 brothers. My second brother is a petrol bank worker. He studied only plus 2, intermediate. Because of family problems, that time whatever decision is taken, I am fortunate enough. I am a scientist. There is no comparison. He is like, I can say, now also I can say, my brother is more happier than me. Really I am telling you, truly, whenever I go to the petrol pump to take his blessings. Because no, we have funded me for my education. So that no, he give some 500 rupees and give it to my wife. Baba నాకు ఎందుకు అంటారు టికెట్ కొనుక్కోమంటాడు అంత ధైర్యం నాకు ఉండదు నాకు 3 లక్షలు జీతం వస్తుంది కొడుకుంటుంది ఇలా చెయ్యి వై ఈ దాంట్లో గుడ్ థింగ్ బికాస్ నో ద కైండ్ హార్ట్ ఈస్ దేర్ ఈజ్ ఎ హెల్తీ వెరీ గుడ్ సో దట్స్ వాట్ నో sir I want to work petrol pump I am not saying that you have to start the petrol pumps to give the employment that time it happened so that no for that we need the education for that what you have to eat is eating I told stress reliever I told good sleep and also to get the good sleep keep walking so there is a Vivekananda proverb I can say when you are happy keep working or walking when you are in stress okay keep walk and working then when you are in the bad mood keep walk and working only two things will always occupy in the works or if no work is there keep on walking పోయి నడుస్తూనే ఉండండి పోతూనే ఉండండి అలసిపోతే మళ్ళీ రండి మొబైల్ ఫోన్ పట్టుకొని చూసుకుంటా అంటే ఐ డోంట్ నో దట్ ఈస్ యువర్ విష్ దేర్ అండర్స్టాండింగ్ ఆఫ్ ద బాడీ ఆర్ ద మోస్ట్ కామన్ లైఫ్ స్టైల్ డిసీజ్ అసోసియేటెడ్ ఫ్యాక్టర్స్ ఆర్ appropriate the health interventions before living and after and all Maiden said when cancer has come we are giving the radiotherapy after that we are giving the what Andas mentioned we are eating the foods also unknowingly we are doing for the things because no just we follow someone will tell in the YouTube take the coconut oil what is the scientific sanctity we keep on drinking good or bad someone will tell no in watermelon they give the injection immediately we will forward to the 10 people without knowing being a biology student we should have the minimum common understanding of what is the mechanism in that really the fruit when you give the injection do it having a circulation system in the body no how it will go we are having the circulation system fruit will be there one dot will be there okay grapes they put in the growth hormones it will grow they put the pesticide on that this is our knowledge without thinking for a second immediately we will send it to the 10 people stop all these things I am telling you go through the books read the books subject is important being a student you have to read it then we can go out of all this healthy diet the lack of physical activity and metabolic changes leads to the raised blood pressure overweight obesity hyperglycemia hyperlipidemia obesity leading to the adverse metabolic effects so lifestyle diseases are in your hand so then keep on walking how many of you are having start from today onwards for good health and better health you are healthy your family is healthy your family is healthy this Bharat is healthy you want to make the Bharat great or not Mera Bharat say loudly Mera Bharat sure huh? you want to become Mahan means start today itself keep your health good when you are healthy thoughts will be healthy when thoughts are healthy you can become a good researcher a scientist again and again telling more than us I want to see you all of you better than me with the knowledge hard work and the skill so for that answer is here what nature is giving then we can start eating and all I am not saying no to others but eat this is also important for you so you are what you eat this is what I can say then before I say thank you save the environment whenever you find the time so in your college lot of places there plant a plant for first years by the time you leave the college in 3 years time it will grow more than us how many of you are having the habit of keeping your college clean and neat are we keeping our college clean and neat when we see the paper no take it and put in the pocket and throw in the dustbin it is our college don't throw anything. Second is, when you go for a plantation, there we can see, we can save the environment. This is the best opportunity in the college time only. Once you leave the college, nothing will come to us. Be happy, be healthy. Wish you all the best. Thank you very much for inviting me. Thank you. Any questions?
Dear students, you can ask many
Anything
questions.
regarding your career also no problem.
Is regarding your food, your thoughts. Actually, sir, no one in my students are obese till now.
That's good.
Future we don't know actually, but the cell phone is blackmailing their brains, their thoughts, and their habits. But we are always trying to check the usage of cell phones. In this we are trying to educate them.
Okay.
They have to become a good educationalist. Then only they can stop the usage of cell phones. That's why we are trying, because all of my students are good students actually.
And even we are
Actually, what is happening? They are good students, but the cell phone is highly evolving and blackmailing their.
you know the pain of sir. You want to you good life, happy life. Mean you have to be educated more. Yes sir, no sir.
Yes, sir.
So that my health is important. I have a small thanks to my younger son. I tell your age group. He put the tambour at my home. At 9 o'clock in the evening, we have to drop the foods. No for nothing. We can talk to mother, or you can go to the newspaper. You can read the storybook. You can read the Bhagavad Gita, whatever book you love to read. Read. They sleep around 10:30. Morning they will wake up before the sun. So that is the 5:30 around. So till 6:30, 7 o'clock they never touch the cell. That is the rule. He put that. I have to follow it. When the phone will come, I will take, but I will not browse. Because no, listen to the good things, whether the younger children are reached brother. పిల్లల పెద్దోళ్ళ మనకు అవసరం అదంతా ఒక మంచి విషయం చెప్పిండా లేదా? డూ యూ హ్యావ్ ద ఎనీ తమ్ముడు ఎట్ హోమ్? మేడం మీ దగ్గర తమ్ముడు ఉందా? హలో, మీరు ముగ్గురు బాగా బిజీ ఉన్నారు. ఎస్, ఐ యామ్ అబ్జర్వింగ్ యూ.
So
Right? No. If you don't put the tambour for you know that you are the cat. You know the cat, common entrance test for the MBA. He got the 95%. He is very unhappy with that. Now I done a blunder mistake. I done a blunder. One. He said, one. Spend more time with my friends. He told very clearly. He is going for the basketball. I am playing every day in the ground for the 2 hours. Where I have to reduce. I did not manage my time properly. That's what I got up. And that is my mistake, he told. How many of you having the guts to say like that? 99% you want, 100% you want to get into the Ahmedabad IIM. The target is, he put the target, not me. I never put any target to anyone. Even my PG students also there. Happy to say, they have the guts to leave. Names scientist are here to the job. Gone the scientist here to the job. Job only say to them, permanent temporary children. One is in the standard. ఎందుకంటే that is very much required. Not only సేమ్ ఉంటుంది ఉన్న మొబైల్లో ఫోన్ పాట పెట్టుకుని వింటుంటారు తప్ప బ్రౌజ్ చేయరు. ఐఎమ్ నాట్ అడాప్ట్ ద టెక్నాలజీ అండ్ ఆల్, బట్ దేర్ ఇస్ ఎ మీనింగ్ ఫర్ ఇట్. వాట్ ఎవర్ అన్నెసరీ థింగ్స్ యు ఆర్ బ్రౌజింగ్, మీరే జడ్జెస్. యు ఆర్ డోంట్ నాట్ బెటర్. ఎస్ సార్. మేడం నాకు ఫోన్ చేసినప్పుడు, సార్ మీకు కావాలి సార్, కొంచెం మా పిల్లలకు మోటివేషన్ కావాలని అడిగింది. ఇస్రాయల్ సార్ ఆల్సో కాల్డ్ మీ. నో సార్, మీ ప్రజెన్స్ ఇంపార్టెంట్. This know what we want is, that is winning yourself. Vivekananda very clearly says, win your mind. Are we winning the mind? Are we winning the mind? have to see what is the happening in my line of research. లేకుంటే వెనకబడిపోతే నేను చాలా రిస్ట్రిక్షన్ rate my projects లేకుంటే లేసర్ IT only upgrade కావాలి. No, what is happening, how they are living, everyone, not only me, all your teachers also, they keep on reading. In the campus, I utilize my 100% time for education. ఒక సైడ్ అన్నం దొరకకపోయి కష్టం.
ఓకే సార్.
చదువుకున్నామా వద్దా? మీరు కాలేజీకి ఎందుకు వస్తున్నారు? నిజమా? చదువుదాం. మీ సార్ చెప్పినట్టు ఈ మనకు పట్టినటువంటి మన భయం అని దీన్ని నాకు లేకుంటే నేను చచ్చిపోను, నేను డస్ట్బిన్లో పడేస్తాను, నన్ను వదిలేమని వదిలేస్తాను, పక్కా వదిలేస్తాను. యూనో మై సన్ సెడ్ నో నానా యాపిల్ తీసుకో అని చెప్పేసి, యాపిల్ వద్దు నానా, 5,000 రూపాయలు మై బడ్జెట్ అని చెప్పిన 8,000 కొనుక్కున్నాను. ఎస్, వై షుడ్ ఐ స్పెండ్? ఈ మధ్య ఆఫీషియల్ కూడా వాట్సాప్ వస్తున్నాయి సార్, తప్పట్లేదు. ఎందుకంటే దే హ్యావ్ టు కీప్ అన్ ఆన్సర్. అంటే మీకు తెలియదు అదర్ సైడ్ ఏంటంటే మేము పార్లమెంట్ క్వశ్చన్స్ ఆన్సర్ చేయాలి, ప్రైమ్ మినిస్టర్ ఆఫ్ క్వశ్చన్స్ వస్తుంటాయి, చేస్తుంటారు. అతను నేను చదువుతుంటారు తప్పదు. సార్ మీకేం సార్, మీరు సైంటిస్ట్, వెళ్ళగానే 3 లక్షల జీతం వస్తుంది మీకు అంటే కరెక్ట్, నిజం, 100% నిజం. మీ వయసులో ఉన్నప్పుడు నేను చదువుకున్నాను కాబట్టి వాడు నా వెనక వస్తుంది, ఎన్నడూ ఉంటుంది. ఇప్పుడు కూడా నేను చదువుకుంటాను. నువ్వు తిన్నా తినకుండా ఉప్పు కొనాల్సిందే అని చెప్పింది. చిన్నమామ జాబిల్లి బొమ్మరి తెలుగు ఇంగ్లీష్ బుక్స్ ఏ పడితే కొంటూనే ఉంటారు. మన బుక్ ఫేర్కి వెళ్తే ఐ లవ్ టు రైట్ ఐ బై బుక్స్. చదివామా? మీ సార్ని నేను మొదటిసారి వచ్చిన మంచోళ్ళగా కనిపించిన సరిగ్గా మంచోళ్ళు సార్.
How many of you wanted to become a scientist in NIN, National Institute of Nutrition, like sir? ఈ సందర్భంలో చెప్పండి, మీరు ఎంతమంది NIN లో సైంటిస్ట్ కావాలనే కోరిక ఉంది మీకు? లేదా? ధైర్యం లేదా? ఆ కోరిక లేదా, ధైర్యం లేదా? సో ఒకసారి వీళ్ళందరినీ NIN తీసుకొస్తాం, అక్కడైనా మోటివేట్ అవుతారో చూద్దాం. ఓకేనా?
రైట్. సార్ ఉద్దేశం ఏంటంటే రాజేంద్ర లాగా సైంటిస్ట్ కావాలి, రాజేంద్ర కంటే ఎక్కువ రావాలని రాజేంద్ర కోరుకుంటాడు. మీ ఏ లైన్ ఎందుకుంటారో ఆ లైన్ ఉద్దేశం అంతే. వాట్ ఎవర్ నేను కూడా ఒక ఫాదర్ని, ఒక వైఫ్, ఒక హస్బెండ్ని, ఒక దగ్గర నేను పనిచేసుకొని వస్తున్నా. ఎన్ఐఎంలో ఒక వర్కర్ని, పబ్లిక్ మనీ తింటున్నాను, కానీ పని చేయాలి కదా, చదువుకోవాలి, నేను పని చేయాలి. నేను మొబైల్లో షార్ట్స్ తీసుకుంటూ కూర్చున్నాను అనుకోండి, ఏమవుతుంది? నేనేం కూడా వెళ్ళగొడతాడు, మామూలుగా కానీ ఇంటి దగ్గర బయటకు వెళ్ళగొడతాయి.
థ్యాంక్ యూ సార్. చాలా బాగా చెప్పారు. చిన్న అంటే టైప్ 2 డయాబెటీస్ మెడికేషన్ అవసరం లేకుండా మనం లైఫ్ స్టైల్ చేంజ్ చేయడం ద్వారా వాకింగ్ మంచి గుడ్ హ్యాబిట్స్ క్యూ చేయొచ్చు సార్.
Right. Question is a type 2 diabetes. We'll bring it back. Madam may answer. Then I can say a pre-diabetic stage లో ఉన్నప్పుడు అంటే వ్యాధి ముదిరగా ఉంది అంటే కదా సార్, when it will not become advanced, we can reverse it. When it will become advanced, then it will start destroying the liver cells. So that now advanced stage నుంచి బ్యాక్ తీసుకురావడం కొంచెం కష్టం. But whereas no pre-diabetic stage, we can. So recently now when I checked my sugar levels and all, so after a long time I saw a pre-diabetic condition. Malli okasari check chestunna. How to reverse it? Because now more sedentary అయిపోయింది. Every day stress is there. If I reach stress నేను feel గాను, it is office work, after office also daily నేను అవుతుంది, నేను వర్క్ చేసుకోవాలి. I have to find skipping on question. నా క్యాంపస్ లో నడుస్తాను, కొంచెం స్కిప్పింగ్ చేస్తాను, నేను మార్నింగ్ లేస్తాను, యోగా చేసుకుంటాను, బయట వేసుకుంటాను, దెన్ ఐ డు ఎవ్రీడే ఎక్సర్సైజ్. దెన్ ఆఫ్టర్ 1 month I check it is normal. We can revert it. It is not because of the many problems, many things are there. It's possible. Better we can add to that.
This is what actually there are differences in diabetes before declaring somebody as diabetic, and the way of writing the article has an impact. So, మీ పేరెంట్స్ కూడా చెప్పండి. మామూలుగా మనం డాక్టర్ దగ్గరికి వెళ్ళినప్పుడు ఒక ఏజ్ వచ్చినాక బ్లడ్ గ్లూకోజ్, HP1 ఇవన్నీ ఫాస్టింగ్ చూస్తున్నారు. ఫాస్టింగ్ ఎంత ఉంది, HP1 ఉందంటే వెంటనే మెట్ఫార్మిన్ రాసేస్తున్నారు. దట్ ఈస్ ద మోస్ట్ idiotic thing the clinicians are doing. They should have a panel of markers before prescribing anti-diabetic drugs. ఏమవుతుందంటే diabetes is a condition where the patient has developed the insulin resistance. Insulin resistance అంటే ఏంటి? ఇన్సులిన్ ఇచ్చినా అదే పని చేయట్లే. ఇప్పుడు ఒకసారి తలుపు తడుతుంది, తలుపు తట్టగానే మనం తీస్తాము. 10 సార్లు తలుపు తట్టాము, అది పని చేయట్లేదు. ఇన్సులిన్ రెసిస్టెన్స్ అనేది ఆ కండిషన్ వచ్చినప్పుడు మెట్ఫార్మిన్ ఇవ్వాలి. అట్లా కాకుండా ఫాస్టింగ్ బ్లడ్ గ్లూకోజ్ చూసేసి వెంటనే ఇస్తారు. సో it's have to be looked at before prescribing metformin. So as you were saying, sir, diabetic, it is an insulin resistance condition. So that is not hyperglycemia, pre-diabetes, insulin resistance. Insulin resistance is not reverse possible.
Pre-diabetic situation we can.
Yeah, hyperglycemia you can. Pre-diabetic you can. Insulin resistance, that is what is diabetic condition.
Thank you.
Every physiologist will bring back, but it may not be 100%.
Please ask questions. Sir is ready to clarify.
Anyone?
Even scholars are there. Swati, do you have a question?
So this test నేను మీ స్టైల్లో ఉన్నప్పుడు మళ్ళీ చెప్తా. మేము లిటరేచర్ విపరీతంగా చదివేవాళ్ళం. మేము క్వశ్చన్స్ అడుగుతాం మా టీచర్స్ని, బుక్ తిప్పుకోకుండా అడగటం. నేను కెమిస్ట్రీ క్లాస్లో ఉన్నా అంటే నేను రియాక్ట్ మా సార్ కంటే 5 నిమిషాల ముందు చెప్తున్నాను. ఎస్, నేను పీజీలో ఉన్నప్పుడు కూడా సార్ నేను తెలుగులో అడుగుతాను, సార్ నేను తెలుగు క్వశ్చన్ తెలుగులో అడుగుతాను. లాంగ్వేజ్ ఏదైతే క్వశ్చన్ అడగాలి చదువుతే కదా బిడ్డ మీరు అడిగే. చదువుదామా? మళ్ళీ అడుగుతున్నా మిమ్మల్ని, చదువుదామా? చదవండి, బుక్స్ చదవండి. పొద్దుగా రోజు 2 గంటలు కూర్చొని బుక్స్ ఇవ్వండి. అలవాటు అయిపోతే 2/4 అవుతుంది. తర్వాత మీరు బుక్స్ ఇవ్వడం ఉంటుంది అయినాక. ఓకే,
like in the game of calories, what is the exact amount of calories a person needs to have a good life?
Okay, రోజుకి ఎన్ని క్యాలరీస్ తీసుకోవాలి, how many calories you have to take. So being an adult, madam nicely showed the plate, my plate. I say my body. Yeah, go through that one. It is recommended in the 2000 kilocalories per day. In that what to eat, how much we have to take, this is what the balance should be said. So for that answer is that no sir, every day we have to balance and eat ante no. So we have to follow that plate. Just to make you understand we have given. Then I said no other remedies there, no how the antioxidants will come and all. We have to listen to someone. YouTube is also seasonal fruits and vegetables తినండి. ఆకులు ఏదో తింటారు, లీఫీ కర్రీస్. Yes, you have to eat it. Then that will give sufficient amount of the things. So don't worry, our food is really having that many calories. Once upon a time we were having the malnutrition and all. Now we are not having that much of malnutrition problem in this country. Now we are having the food in front of us. But choosing is a problem for us. Just I am not going to give the details to you because my wish is you have to read and go to the NN website and see. Already I have explained the live date. RDI and recommended dietary allowance and book ఉంటుంది, free to download it. My only free books. Now government is made everything is a free. Indian government, the Bharat government is taking the steps to give everything is a free. మీ కెపాసిటీ. మేము చదువుకున్నప్పుడు ఒక బుక్ కావాలంటే కొన్నాడుతున్నాం. ఉమా మేడం తీసుకోవాలి, ఆయన తీసుకోవాలంటే సార్ సాటర్డే మీరు తీసుకోండి, సండే నేను తీసుకోవాలి, బదిలాడుకుని పోతున్నాయి. మీకు అటువంటి బాగలేదు కదా. Okay, that is 2000. Yeah, correct.
Now the balanced diet is available for all students. Now it is the time to go for a beautiful lunch for all students.
Now I request this technical session chair, Professor 𝕃𝕀𝕍𝔼 to sum up the session. On behalf of organizing committee, I extend our sincere appreciation and we are very much thankful to you, sir. Madam, please.
So on behalf of the organizers, I thank Dr. Rajendra, who has been associated with NIM for a long time. ICMR NIM is such an institute which has been pioneering to work in diabetes particularly and the lifestyle related things to diabetes. And Rajendra was such an active researcher and known him for the past several decades, I can say. So such an active, humble person. Personality was such a humble person and always helping. That is my personal opinion. Coming to this session, really I'm thankful to you, Dr. Rajendra. So to whatever extent that I can understand, you have put the contents of your talk in a nice manner, which everybody can easily. Though you are presenting your works at very high scientific platforms and research platforms,
is the greatest thing that you have done with your work. Thank you very much, Dr. Nazira.
We are very fortunate enough to have you here, sir. Apart from the scientific talk, also given motivational talk to our students. Now it is the time for felicitation. I request the faculty of Department of Zoology to come out to the dais.
Here from this college, my student Kishore is working here. Biotechnology. Right? And also, Vasi is known to me, Samati Madam is known to me. So, he is here. And also, all your faculty is now my. Thank you. All of you are my friends. You can feel free to write to me. Thank you.
Now this technical session is completed. Now it is the time for lunch. Now the time is 1:30. We shall take 1 hour time and come 5 to 15. Take 45 minutes only. Come to the hall by 2:15. Yes. Students, go to lunch immediately. Have lunch and assemble here in the hall by 2. Side team, please welcome madam. Now I request Dr. Gauri madam to introduce our speaker of this session.
Thank you, ma'am, for accepting our invitation, spending your valuable time. Come all the way from university despite your busy schedule to educate our students and to motivate our students towards research. I will read out a brief biodata of Madam, though she had a lengthy biodata. She has teaching experience. Professor G. Shamita ma'am has teaching experience more than 25 years. She has research experience for about 25 years, and she has done major research projects, and she has guided 11 students which are awarded with the doctoral degree. She had many publications and proceedings. She has written 14 books. She has published many research papers in national and international seminars, conferences. She has 9 foreign presentations, 22 national conference presentations. She has taken up several administrative activities. She worked as NSS Program Officer for Women. She has received National State Award in 2021. She organized 12 national level conferences. She has been awarded with Fellowship of Telangana Academy of Science, Geological Society of India, Indian Science Congress. She played administrative roles as Head Department of Geology, Chairman Department of Geology, Biochemistry, Sericulture and Environmental Science. She worked as IPAC Coordinator for the NAC, which was held 2 years back. She worked as Dean and also Academic Audit. She worked as a Director, Center for Women Studies, and she was in charge for University Library, and this is her present role. I invite ma'am to deliver an elaborate talk on animal models. Madam is going to deliver a talk on role of animal models in biomedical research. I welcome ma'am to deliver the lecture.
Thank you, Dr. Dr. Swami, for introducing me to the August gathering. So today I am very much honored to be with you all today. The new young budding graduates from Kakatiya Degree College, which is a very prestigious degree college in Unforgettable. I congratulate you all for being a part of this international seminar, because organizing an international seminar is not a joke. It is a very big proceeding in itself. It involves a lot of communication, it involves a lot of invitations, correspondence, and it cannot be happened without teamwork. So I congratulate the staff of Department of Zoology from JVC. I'm heartily encouraging them to conduct many more such seminars. At the outset, I congratulate all of you, Dr. Swami, Dr. Ruby and Madam, Dr. dronevideo. So they were all pursuing PhD in Kakatiya University Department of Zoology. So there they were students for us, but now they are honorable teachers for all of you. We are very proud of them at the outset. And when they are conducting such seminars, it is profound duty of us to congratulate them, to encourage them, and also to participate. And I thank them for making me a part of this seminar. Thanks. So the title of the seminar is Advances, Opportunities and Challenges. It is a very big topic actually. And it's a very senior most he is sitting at the back side, so I could not see him because of this flash of lights. Dr. Dinesh sir is my senior actually, he is my senior. I thought he is the principal of this college because he has a lot of experience in teaching and administration. He is a silent supporter for all the academic and administrative activities which are taking place here, I suppose. Yes, sir. But he will not acknowledge anything. He will not take any credit. He is very humble and very grounded to earth. And because of my PhD work, I knew him. He was a senior in our lab. We have completed our PhD under the guidance of Professor 𝕃𝕀𝕍𝔼 sir. So he was my elder brother there. So in research, we call seniors as brothers and juniors as younger sisters or younger brothers. So like that, we are all academically related in one family, that is zoology family. And in my PhD work also, I have been from Dr. Dinesh sir. Thank you very much sir for involving me in this seminar, and I am very proud to be a part of this. So seminar topic, as I told you, it's a vast topic, medical research. But we are only zoology students. So whatever position we are in, whether it is professor or scholar or teacher or whatever, we are all students always. Okay, from you to us, we are all students. So, as zoology students, we work only with animals. Zoology means what? Study of animals. So, biomedical research involves mostly it is human beings. It is final goal is treating human beings. So, in that human being treatment and therapies, animals are used. Okay, so that is my topic, today's topic. So, role of animal models in biomedical research, a review. So, Dr. Rohini was asking me, why madam review? You can tell your work no. So, because it is only a review, because this work is not conducted in our lab. So, that's why I collected a lot of information to introduce you to animals who are playing in the movie of biomedical research. I know when I say movie, you will get connected. Is it not? You all watch movies, right? So biomedical research is like a very big topic, and in that, use of animals is it? Introducing, because we belong to zoology, so our role is to speak about animals. I have taken up animals which are used in biomedical research. So what is biomedical research? So definition. So for any subject first we have to take the definition first. Biomedical research is a multidisciplinary field integrating life sciences and physical sciences. So life sciences means you all know it is botany and zoology, life sciences. And it integrates with other subjects also like mathematics, computer science, algorithms and many other subjects. So all this is the era of bioinformatics. So in olden days all the subjects were separated, divided, but now all subjects are merging to form bioinformatics. So this is one biomedical research is also one of the wings of bioinformatics. And one more thing is it is amalgam of omics. Amalgam ante telusa? What is it? Anybody? Amalgam means it is a mixture. Okay, it is a combination. It is a combination of omics. Have you heard about epigenomics? So all these omics are also combined in this science. So it involves basic science, clinical studies and all omics like proteomics, genomics. So in the structure, in the diagram you can see this is taken up, this is taken as a reference to show you from big data which was published in current research in biotechnology. So it involves genomics, proteomics, transcriptomics, and it is pipelined by the computational data. So all these studies, the data from all these omics is combined and it is studied by computational skills and techniques, and finally, and all this is very costly affair, it is not easy. That is why we are unable to do here. It is happening in many big laboratories which are heavily funded. It's a very costly research. So, and finally its social impact is, it is helping human beings in the treatment of diseases. So before treatment, it has to be identified what disease it is. Then after identification, it has to be treated. That means therapy has to be done. So once the treatment is successful, then that particular drug is discovered. That is called drug discovery, which is done by pharmaceuticals. Okay? So many people are involved. Animals are involved, then pharmacists are involved. Animals like even human beings are also sometimes used as used for clinical trials. So finally, social impacts like drug discovery, then marketing of the drug, and finally utilization on human beings. So steps in drug development. So you can see the red arrow marks there. First, animal models are taken. So in biomedical research, the first step itself is utilization of animal models. So then therapeutic strategies. So how this drug has to be given, all those strategies. Then finally a drug will be developed, and after the drug development, it will be tried on human beings, clinical trials. So when human beings are being tried, so generally in KMC and all, when we go for meetings, they will discover drugs and they will write projects. For that, for clinical trials, human beings are taken. So human beings, they will take their consent. So this is a new drug. Do you want me to apply on you? So they will ask, the doctors will ask. So if the patient says yes, he will sign and then only the drug will be given. So that's called clinical trial. So finally you can see how many animals are used, from cockroach to final cockroach. Then can you identify those animals? What are they? Cockroach, fish, bird, rabbit, monkey, and finally human being. So these are the several steps for trials of drugs. So drug discovery by animal models, as I told you, drug discovery, then pro-clinical research, then clinical development, clinical approval. So once this drug is successful, then it is approved by clinical approval committee, and then it is entered into the market. Finally, you can see pre-clinical development. All these are the various steps which I am skipping because you know what is the difference between in vitro and in vivo? Yeah, you can say you can say okay. In vitro means what? In vivo means what? In vitro, the experiments will take place inside a test tube. In vivo means the experiment will take place in a living organism. It may be animal or human being also. In vivo and in vitro. Then key applications in research. So only I will, you all follow the bold step. Drug development, bold words. Drug development, disease modeling, testing, and surgical innovation. So this, all these are various steps which are involved in biochemical research. So here I have given one slide, CRISPR-Cas9. This is a very new technology. One day your Swami sir will take the class on CRISPR-Cas9, I think. Because he is very well educated, he keeps on updating himself. I observed him during his research tenure. So one day he can explain everything about CRISPR-Cas9. This discovery, it has taken the world by storm. The DNA discovery in 1953, the DNA discovery, it has taken the world by storm. DNA discovery structure ananta telisipoyinaka world lo undeటువంటి genetics vidhanalu mottam maaripoyayi. It has taken the discovery. So CRISPR-Cas9 also has taken the medical research by storm. So what is the importance of this technology? So here బ్యాడ్ హ్యాబిట్స్ అట్లా గుడ్ హ్యాబిట్స్ ఉన్నాయనుకోండి, నువ్వు ఎందుకు మారవురా అని ఎవరో ఒకళ్ళు అంటారు. నేను మారను నా డిఎన్ఏలో ఉంది అంటారు. అంటే డిఎన్ఏలో ఉంటే ఇక మారనే మారు కదా, కానీ CRISPR-Cas9 ద్వారా మార్చొచ్చు. ఓకే, సో ఎప్పుడు మారుస్తాం, దానికి కూడా టెక్నిక్స్ ఉంది. నువ్వు ఇష్టం వచ్చినట్టు మార్చరాదు, దానికి కూడా కమిటీలు ఉంటాయి, అన్నీ ఉంటాయి. సో ఇఫ్ దేర్ ఇస్ ఎ డిసీజ్ లైక్ HIV, అంటే పెద్ద పెద్ద ప్రాణాంతకమైన డిసీజెస్ లోనే ఇలాంటి టెక్నాలజీ వాడతారు, అన్నిటికీ ఏం వాడరు. ఓకేనా? సో CRISPR-Cas9 is a technology which is a molecular technology by which viral diseases, very dreadful viral and bacterial diseases, they can be cured by cutting the DNA sequences in the normal DNA. Normal DNA will be there, then the disease, because DNA of bacteria or virus integrates means mixes with human DNA and it is causing disease in our body. So this technology cuts and how many strands are there in DNA? So 2 strands, double stranded. So by this technology, the 2-stranded, double-stranded DNA can be cut wherever this erroneous DNA is there, and because of this Cas9, which is a protein, so it can cut the DNA at both the strands and it can create a new DNA sequence. So that is the main, only gist I am telling you, but one day sir can tell you in detail by giving examples and all. So that is the main application of biomedical research. Now it is the latest technology, CRISPR-Cas9. So CRISPR is it is an acronym. Its full form is clustered, regularly interspaced, short palindromic repeats. Okay. Then finally, I am entering into my own topic, that is use of animals. So scientists use animals. Ivanni technologies ivanni cheyali ante oka experimental basis kavali manaki. So for that, animals are used. Use of animals to learn about health problems. So when we are using animals, We have to use it for correct purpose, right purpose. So to study diseases, we have to use animals. So why animals are used? Why not plants? Because, yes? Because animals are used because they are similar to human beings in many aspects. See, all animals and plants, if you take and if you go deeply into the molecular level, both of them are having DNA. Both of them are having in DNA. How many nucleotides are there? How many? What are they? Adenine, guanine, cytosine, and
They are saying
thymine. Okay, so whether it is plants or animals, all at the molecular level, all are same. All of us are having DNA with 4 nucleotides. Okay, but with in relation to physiology and anatomy, animals are close to human being. Human being is also an animal. The experimental animals which we are using, they are very much close to human beings. They are similar to humans. They also have same health problems. That is why we are taking them as models. So you can see why are mice used in biomedical research. Have you ever heard about mice being used as research models? Okay. So mice, their anatomy and physiology are same. In earlier days, that means when we were students of intermediate, we used to have dissection of cockroach, earthworm and frog in our intermediate. Did you have? Yes, ma'am. Did you have dissection? Yes, ma'am. Just theoretical or practical?
virtual dissection.
Virtual dissection which was shown in computers. Is it not? But when we enter into MSc, our predecessors had a pigeon also, pigeon dissection. So nowadays all dissections are gone. In rats, rabbits also they used to dissect in the olden days. But because of animal ethics committees are coming, but animal protection committees are coming, so we are not supposed to dissect any animals. So it is all guided now. So you see, rat is similar to human being in anatomy and physiology and also genetics. And they face the same health problems. But why we are using it for research purpose means just like pea plant in Mendel. Mendel's last pea plants, Pisum sativa. Why he has used Pisum sativa? Yes? Its life cycle is short. Within 20 days, your seed can germinate and it can grow and it can also yield flowers and fruits, that means peas. So within 1 year, 4 to 5 crops can be raised. So just like that, even rats also, they are also small in size, easy to handle, and they reproduce very fast. So that is the reason mice has been used for biomedical research. Short lifespan, smaller size. So you cannot breed elephants, is it not? Can you breed 10 elephants in front of your house or in front of Kaktiya University? Because it is very costly. But rats you can grow or you can raise them in a small laboratory because of their small size and high reproductive capacity. And scientists can easily control the environment. Okay? So you can see the scientist who is growing labs in different environmental conditions. You can see several blocks are there. So each one is having different temperature, different humidity and different environmental conditions. So in one room only, he can grow several types, several rats with different environmental conditions. So that is one reason why these small animals are chosen for medical research. And one more thing is, instead of direct animal, nowadays in vitro studies has developed a lot, and there is a new technology called organ on a chip. It is like if you want to study kidney, then you need not sacrifice whole rat. Only one kidney you can dissect and put it on a test, put it on a slide, and then you can do the rest of the experiment. So that is called organ on a chip. So you can see in the circle, kidney, brain, liver, heart, all these organs are there. And small, not even full organ need not be taken, only small. You can just take a small piece of the tissue and you can study on the chip. Chip means not ordinarily our microscopic slide, but it is a very well established with a lot of facilities, with a lot of features will be there on the chip. So you can see the structure of a chip here, how broad it is. It is having a capillary channel, it is having vacuum, and there you can see the structure of kidney with porocytes. Structure of nephron, you can see porocytes are there. So that is called organ on a chip. And for taking the animals for biomedical research or any research, there are several guidelines. So these guidelines were first formulated by two scientists, namely Russell and Burke, way back in 1950s or somewhere in UK. They were the first scientists who said we should not sacrifice animals for everything. So they have written a book called The Principles of Humane Experimental Technique. మీరు అందరూ డ్రైవింగ్ చేస్తారా? డ్రైవింగ్ ఏదో బైకో సైకిల్ కారు ఏదైనా ఒకటి డ్రైవ్ చేస్తారు కదా. సో డ్రైవ్ చేసేటప్పుడు ఎట్లా డ్రైవ్ చేస్తారు? ఒక హ్యూమన్ టచ్తో డ్రైవ్ చేయాలి, హ్యూమన్ బీయింగ్ లాగా డ్రైవ్ చేయాలి. మనం వెళ్ళేటప్పుడు చిన్న పిల్లలు వెళ్తుంటారు, స్లో చేయాలి. పెద్ద మనుషులు వెళ్తుంటారు, స్లో చేయాలి. లైట్ పడుతుంది, ఆపాలి. అంటే నా ఇష్టం, నేను పోతారా పోగలరు. హ్యూమన్, దట్ ఇస్ కాల్డ్ హ్యూమన్ డ్రైవింగ్. ఒక మానవీయత భావంతో చేసే డ్రైవింగ్. సో హియర్ ఆల్సో వెన్ we are sacrificing animals, humane principles have to be followed. So that book was written by William Russell and Burke, who has postulated three main principles, that is 3 R's: replace, reduce, refine. Okay, replace means using non-animal models. ఎంతో అవసరమైతే తప్ప యానిమల్ని వాడదు, అదర్వైజ్ వితౌట్ యానిమల్ వాడేది ఉంటే ఆ వర్క్ చేయాలి. యూజింగ్ నాన్ యానిమల్ మోడల్స్, సెల్ కల్చర్స్. జస్ట్ నౌ ఐ టోల్డ్ యూ కెన్ ఎక్స్ట్రాక్ట్ సమ్ కిడ్నీ సెల్స్ ఆర్ బ్రెయిన్ సెల్స్ అండ్ యూ కెన్ డూ ద వర్క్. దెన్ సిమ్యులేషన్స్ అని ఇప్పుడే వర్చువల్ డిసెక్షన్ అన్నారు. అంటే వర్చువల్ అంటే కంప్యూటర్లో ఒక ఫిష్ క్రియేట్ చేసి దానికి నర్వస్ సిస్టమ్ క్రియేట్ చేసి ఆ నర్వస్ సిస్టమ్ని మనం డిసెక్ట్ చేస్తాం. వర్చువల్, ఇట్స్ నాట్ రియల్ ఫిష్. Okay, so ఎప్పుడైనా మీకు చూపించండి వర్చువల్ డిసెక్షన్. ఓకే, సో దే విల్ అండర్స్టాండ్. సో వాట్ ఈస్ రియల్, వాట్ ఈస్ వర్చువల్. రియల్ అంటే నిజం ఫిష్ని డిసెక్ట్ చేస్తాం. వర్చువల్ అంటే ఒక ఫిష్ని డ్రా చేసి ఆ ఫిష్ కదిలేటట్టు చేసి కంప్యూటర్ టెక్నాలజీ తోటి సో దాంట్లో మనం డిసెక్షన్ చేస్తాం. సో దట్ ఈస్ కాల్డ్ వర్చువల్. సో సిమ్యులేషన్స్ అంటే అదే. సిమ్యులేషన్స్ మీన్స్ imitations. Then reduce, instead of 5 rats use only 1 rat. So reduce the number of animals. Next, refine. So improve, so refine means what we parchable. So you have to use chemicals or harsh chemicals we are not supposed to use. Improving procedures to reduce pain. Even animals, they feel pain. So మనకు పెయిన్ ఉందంటే కదా వాళ్ళు మనం ఒకలాగా ఉంటే మనే కదా మనకి సాక్రిఫైస్ చేసి మనకు పెయిన్ ఉందంటే వాళ్ళకి కూడా పెయిన్ ఉంటుంది. So that's why we have to reduce pain. So and improve animal welfare. So we have to take care of them. We have to handle them gently and with care. So that means refine. So this was published in John Hopkins Center in UK. So apart from this, every country has some guidelines, some rules for animal handling. So you can see in 1876, just like this earlier two scientists, in 1876 itself, Prevention of Cruelty to Animals Act was published. And later on, several countries like France and Europe and all countries have their own ethical committees, including ours, that is India. In India, in Hyderabad itself, in National Institute of Nutrition, National Institute of Nutrition Policy. So, androne oka center undi, NECLAS, that is National Center for Laboratory Animal Science. Okay, so this institute has given clear guidelines. So those guidelines are called SIPC. So there you can see Committee for the Purpose of Control and Supervision of Experiments to Animals. So this SIPC, it is a very big book. You can see there is a big book having guidelines for handling animals. So we have to follow those guidelines before we touch any animal. And finally, animal research saves lives. ఇంత కష్టపడి మనం ఎందుకు చేస్తున్నాం ఈ పని అంటే మన జీవితాలు ఇంప్రూవ్ కావడానికి, డిసీజ్ నుంచి నార్మల్ కావడానికి, లాస్ కాకుండా ఉండడానికి. Animal research saves lives. So in what way animals have helped us? So you can see major discoveries are insulin for diabetes. Pig insulin తీసుకున్నాను, Chana mandi diabetic. India 70% above people are diabetic. Now it is a diabetic boom in entire country and also in the world, all over the world, because of the food habits. Okay, so insulin mana production, insulin ekkada produce avutundi body lo pancreas, very good. So మన ఇన్సులిన్ సరిపోతలేదని యానిమల్స్ లో ఉన్న ఇన్సులిన్ మనకి ఇస్తున్నారు ఇంజెక్షన్స్ ద్వారా, షాట్స్, ఇన్సులిన్ షాట్స్. You must have seen your elder people or somebody who is taking insulin injections. Have you seen anybody taking insulin shots? So that insulin is taken from other animals. Can other human beings donate their insulin? Can you donate? Can I donate? Already I am having less insulin, so I am taking tablets. So if it continues, I have to take from insulin from other animals. Blood ఇచ్చినట్టు ఇన్సులిన్ ఇవ్వరు, బ్లడ్ డొనేషన్ చేస్తాం మనం. ఇన్సులిన్ డొనేషన్ కష్టం. Okay? So insulin was taken and it was tested on dogs first. How dogs are reacting. Okay, penicillin is an antibiotic. It was tested on mice. COVID-19 vaccines, we are all witness to that history. పెద్ద హిస్టరీ అయింది. ఎప్పుడైంది COVID-19 to 21 మధ్యలో ఉన్నామా మనం? ఉన్నాం అప్పుడు? ఎక్కడో అయింది, ఏమో అయింది, చూడలేదు. పోలియో అయింది ఎప్పుడో 1970s మేము పుట్టినప్పుడు, కానీ అది చూడలేదని మనం ఓకే we have read somewhere. But COVID, we have all seen how COVID was treated with the help of vaccines. So that was first tried on monkeys, and Hyderabad was a major place where COVID vaccines were prepared. So blood transfusion techniques, మనకు blood మన A group వాళ్ళు B group వేయదు, B group వాళ్ళు వీళ్ళకి వేయదు అని తెలుసు కదా. బ్లడ్ ట్రాన్స్ఫ్యూజన్ టెక్నిక్స్ మీకు చదువుకున్నాను ఆయన కూడా నాకు తెలిసి ఉంటున్నాడు, సైన్స్ స్టూడెంట్ లాంగ్వేజ్ వస్తున్నాడు. సో all these blood transfusion techniques were studied in dogs, cats, horses, cattle, and all these models. So these models enable the research to understand disease mechanisms and treatment safety before human trials. Human being ki chese mundu pratidi animals ki chestunnadu. So as I told you, several achievements are there in discovery of vaccines, diabetes treatment, antibiotics, cancer therapy. Lot of research is going on with regard to cancer, heart diseases and brain diseases. So phylogenetic relationship. Have you heard about phylogenetic genetics subject ఉంది కదా మీకు? Phylogeny అని విన్నారా? Phylogeny means it is a tree. ఒక ట్రీ చెట్టు ఫస్ట్ వేర్లు ఉంటాయి, తర్వాత ట్రంక్ ఉంటుంది, తర్వాత బ్రాంచెస్ ఉంటాయి కదా. అదే విధంగా ఇది కూడా ఫైలోజెనెటిక్ ట్రీ. అంటే ఇవి రిలేషన్షిప్ చెప్పేటువంటి ట్రీ, ఫైలోజెనెటిక్ ట్రీ. సో ఎవ్రీ ఫైలోజెనెటిక్ ట్రీ హ్యాస్ సమ్ బేసిస్. ఎప్పుడు మీరు ఎంఎస్సీకి వెళ్ళినప్పుడు మీకు జెనెటిక్స్ ఎవల్యూషన్ అట్లాంటి టాపిక్స్ వచ్చినప్పుడు ఫైలోజెనెటిక్ ట్రీస్ చాలా రకాలవి లేబులింగ్ చూడాలి. ప్రతి ట్రీ కింద ఒకటి దిస్ ట్రీ ఇస్ బేస్డ్ ఆన్ అనే వర్డ్ ఉంటుంది. ఓకే, బేస్డ్ ఆన్ ఫైలోజెనెటిక్ ట్రీ. సో ఈ ఫైలోజెనెటిక్ ట్రీ ఒక ప్రోటీన్ మీద బేస్ ఉంది. Okay, so that protein is called as ADAMTS2. I will give the details in the next slide. So based on this protein, you can see human being, cattle, rat, frog, zebrafish, nematode and fruit fly. Fruit fly and Drosophila. They are all related. ఇదన్నీ కలిపి branches. Branches కనుక trunk కనుక ఒక root ఉన్నది. ఎక్కడ animal, human being, ఎక్కడ Drosophila, fruit fly. They are related based on, in all phylogenetic trees, observe it is based on. Suppose based on our pancreatic discharge, that is insulin. Based on insulin, మీకు ఒక ఎగ్జాంపుల్ చెప్తాను. హ్యూమన్ బీయింగ్ మూడు ఇస్తాను. హ్యూమన్ బీయింగ్, మంకీ ఆర్ చింపాంజీ, ఏదో ఒక ప్రైమేట్, హ్యూమన్ బీయింగ్, చింపాంజీ, పిగ్, 3 animals ఇస్తున్నాను. Which two are closely related? Human being, chimpanzee, pig. Which two are closely related? Human being, మనిషికి ఎవరు దగ్గర? చింపాంజీయా, పిగ్గా? Primates, they come under one family, primates. Yes? So you are saying pig in what way? You have any basis? No, if you are correct actually. Based insulin.
Actually, kidney transplantation.
Yes, based on at the Pratidan, so kidney is based on kidney and chapala, phylogenetic tree. That is what I am coming to the point. So based on insulin, pig is more close to human being than chimpanzee. Than insulin kante pig insulin manaki ekkuva panichestundi. Okay, so for everything there is a based on in phylogenetic tree. So based on ADM, TS2 gene and protein. So this is the phylogenetic tree. So this is the protein ADM. It is a disintegral metalloproteinase. It is a very big word, and its main function is to control the levels of this what is that, collagen, pro-collagen, which helps in strength, support and elasticity to body tissues. So this protein is found not only in these 5 organisms. This protein is found in nearly 285 organisms. Okay, so based on this protein, the phylogenetic tree for 5 animals was created, because those 5 animals, they are mostly used in biomedical research. So commonly used animal models in research. Just now in the phylogenetic tree I have given these same animals like mice, rats, zebrafish and ferrets, also some mammals. They are burrowing animals. They are also used. They are very small animals. So they are also used for biomedical research. So here in the phylogenetic tree you can observe same animals. Okay. So these are the main animals which are used in research. So why these animals are used? So you can see Drosophila, you see, if you study genetics book, intermediate genetics, intermediate genetics BSc which book you are following? All in one Telugu Academy, all in one. Adena different books follow
different books.
Reference books. Yeah, in reference books, first book which has to be followed in genetics is 𝕃𝕀𝕍𝔼. Genetics by 𝕃𝕀𝕍𝔼. Genetics నుంచి ఫస్ట్ పేజ్ లాస్ట్ పేజ్ వరకు అన్ని కాన్సెప్ట్స్ కరెక్ట్గా ఉంటాయి. సో దాంట్లో మీరు అబ్జర్వ్ చేస్తే ఎవ్రీథింగ్ ఈజ్ బేస్డ్ ఆన్ డ్రోసోఫిలా. మొత్తం డ్రోసోఫిలానే అందరూ. Metabolism. Have been studying in these organisms. Then rabbit is also studied for studying Alzheimer's disease of remembrance. And then equids, horses are also they are also used, but maintaining horses is very expensive. And other models like cattle for pregnancy related issues and reproductive analysis. Cows are taken. From goats are they are used for orthopedic models, orthopedic diseases and all. Sheep for all these wound healing purposes. grafting occurs during operations also, major surgeries. One part of the skin is removed from a healthy part and it is patched onto where heart surgery or kidney surgery it has taken place. So how the patches. So that is first studied in all these animals like rats, bone models, grafting, and animal models for tuberculosis. If we go on studying, we can take number of diseases, thousands of diseases are there, but I have given only few diseases commonly occurring and most popular diseases I have taken. So here the tuberculosis, all these animals were used for studying tuberculosis. In olden days, TB patient because we didn't know the treatment. Now treatment is there. Regular courses are given. And before this treatment was given, all these animals were used for studying this TB studies, and now TB is treatable. It is treatable. So animal models which helped us in TB treatment. Animal models for other diseases like grafting, disc degeneration, like bones, all the vertebral discs. And cancer, it is still going on. It is a very big subject. Cancer, role of animal models in cancer therapy. So not only animals directly, but tissues have been taken because cancer is nothing but a tumor. A tumor can occur in any part, like it may be occurring in blood or soft tissue or bones. So there you can see the tissues were taken and the studies were taken up. So this is the reference pivotal role of preclinical models in anti-cancer treatment. So tissue regeneration for intervertebral disc. So this is also all these animals were taken. You can see the backbone. So in those backbone of the animals, this research was conducted. So next is invertebrate animal models. So far whatever we have discussed, like cat, cow, dog, all these are higher animals, mammals, vertebrates. Okay, fish is a species, frog, amphibian. So they are all vertebrates. Okay. So here you can see these are invertebrate models. Drosophila is a whether vertebrate or invertebrate. Drosophila. Vertebrate. Invertebrate. It is an insect. Arthropoda. Okay, so there are several invertebrate models. You can see such as nematodes like 𝕼𝕝 elegans and chinna nematode, microscopic, only 1 mm in size. So it is used for embryological studies. So insects, freshwater and marine planarians, Vijaysia, crustaceans like arthropods, molluscans, all these are also animal models for medical research. And unidentified top invertebrates were Drosophila melanogaster and 𝕼𝕝 elegans, which is a platyhelminth. So here you can see these two animals. I have given the picture. Drosophila. Actually, not Drosophila, but if you zoom it, it looks like that. Okay, and 𝕼𝕝 elegans, I have given you male and female birds. They are only 1 mm in size. It is a microscopic picture. Okay, so these two are. I have given several references were studied on all these diseases. You see, all the systems, anatomy, physiology, everything was studied in these two animals, which are invertebrates. So coming to my own family, that is silkworm. Swami is a part of our family that we did silkworm research. Okay, and this we all come from Mangla, so we work in entomology. Okay, so silkworm is our animal, and silkworm is an excellent model, invertebrate model. What is the speciality of silkworm? Silkworm, it yields silk. That silk we are wearing. Human beings will not give any, even, what is it? It is like a chima doma, silkworm. Other products, lac, honey, silk, all these are insect products we are using. Okay, so it is just like our Mendel's pea plant, silkworm is also having very short lifespan, and within 1 year, 5 to 6 crops we can grow in one room, and we can have thousands of moths at a time. So because of that high reproductive rate, silkworm is taken as a model. And and these are the several biomedical applications of silicon. Just like how higher organisms are having several applications, silicon also has all this metabolic and disease research, then diabetes, gout, and all other diseases which we studied earlier, they were all studied in silicon. And what is the advantage of invertebrates is, here especially silkworm and other insects, we don't have any ethical rules. Our book follow there is no requirement for following ethical rules. So that is the main advantage, and they are cost effective. They are very cheap. They are not expensive to maintain. So that is why silkworm is taken. It is very simple, and we need not follow ethics for that. And several advantages, as I told you, the environment safety, and it also looks very beautiful when we are wearing silkworms. We will be having a garden, beautiful mulberry garden will be having, it will yield fruits. Mulberry fruits, berry fruits are very good for health, which having vitamin B and all those complex vitamins which cure many diseases will be there. So growing silkworm is a beautiful thing because we are having a garden with flowers, with fruits, and with butterflies. Okay, when fruits are there, automatically bees will be coming. So it is a good life doing sericulture. So we have several advantages. Environmental safety is there, and they are also less toxic. Honey bees ni pencham anukondi, are very toxic. Honey bees ni pencham kutte, maname kutte. Manam poi danni honey kavali ani, techina honey kavali ani poyar anukondi. Adi manam kutte. Silkworms kuttavu. Silkworms are very safe. Ask your sponsor how many silkworms has read. He filled our lab with silkworms. They are very harmless. They are not toxic. Okay, so that is another advantage. So this is our, because whatever we do, we have to show you, because the Kakatiya University of Biomedical Research changed, but we started with silkworm. చిన్న silkworms ని పెంచి మేము కూడా బయోమెడికల్ రీసెర్చ్ కి మా వంతు కృషి మేము చేస్తున్నాం. సో డిపార్ట్మెంట్ ఆఫ్ జూవాలజీ Kakatiya University is rearing 𝕃𝕀𝕍𝔼. 𝕃𝕀𝕍 is a very big sized silkworm. Okay, and we have a different garden for that, terminalia plantation. You can see all these silkworms. This last one, this is our own silkworm, and that is the picture showing the cocoon. And cocoon, it is made up of silk fiber. In the second set, you can see one silk fiber, which is a scanning electron microscope picture. It has two fibrils in them, sericin and fibrin. So this sericin and fibrin, they are biomedically important. Sericin is the cementing substance which covers the double fibrils, that is fibrin. So this is the significance of fibrin. Fibrin is helpful. Nowadays, a lot of research is going on in tissue regeneration. In that, fibrin is used. So these are the multiple applications of fibrin, biomedical applications of fibrin, drug delivery, and even after operations in surgery, silkworm threads are used because they are biodegradable. Other threads, they have to be removed. But whereas if the stitches are done with silkworm thread, it is biodegradable. Harsh treatments are not utilized for removing the stitches. So they are biodegradable automatically. They will mix up with your tissue because they are also made up of organic material. So gene therapy is also going on in that. So you can see wound dressing, films, porous sponge, hydrogel, 3D scaffolds. They are all prepared from fibrin, silkworm fibrin. So we have started our work with fibrin extraction in our laboratory. We have done silkworm fibrin extraction. Then we have done the studies on their properties. Then scanning electron microscope studies also we have done. And our results have shown that this Tassa silkworm silk has several properties which are similar to Bombyx mori, and
capacity and tissues development, elementary canal, fat in that animals. మనకు కాకతీయ యూనివర్సిటీలో ఒక కార్యక్రమంలో మా ఫీల్డ్లో that is very very beautiful insect. అంటే మనకు బాగా అలాగే లాబొరేటరీకి వచ్చేది, దాన్ని తీసి మన వర్క్ అంతా కంటిన్యూ చేసుకునేది బయోకెమికల్ వర్క్ కూడా. యాక్చువల్లీ I have enjoyed with the 𝕃𝕀𝕍𝔼 silkworm biochemical work. Even though molecular work we have done, in the molecular work we isolated the proteins. Then the work is stopped in the 2022. The madam awarded me as a doctorate. So thank you madam on this days. We met on this days. My dear students, I give you lot of freedom to you to study. Actually, I have learned this from madam. Academic freedom, because for academic excellence. When there is a freedom, there is a excellence. That is I learned from the madam. Thank you madam.
Good afternoon. We had the opportunity of listening to Professor 𝕃𝕀𝕍𝔼. She was once a head, now she is the Chairman Board of Studies of Zoology Department, Kakatiya University. Thank you, madam, for accepting the invitation and sharing your knowledge and the sentinels of research in zoology. So, madam also belongs to the same entomology research lab where I was awarded a PhD. And coming back to our present lecture, madam gave us a pictorial depiction of how the animal is used for the benefit of humans. We are the apex animals in the phylogenetic tree, and we are using lesser grown animals for our own benefit. And madam work mainly on silkworms, and the other important use of a silkworm is the silk thread. Madam elago chepparu meeku how the silk thread is being used. Akkada oka important strategic defense related research in that now they are using silk thread to make bulletproof jackets. Earlier our soldiers were using steel jackets and they were costly. We have to import them and they were very heavy. Each jacket used to weigh not less than 15 kilos. A soldier has to bear 15 kilos apart from his stick pack and a gun. And he has to walk knee-deep snow in Himalayas to protect us. So we are very happy that we are enjoying our lives here. So when we use an anti-bulletproof jacket made of silkworm thread, it won't be more than half a kilo of weight. So a soldier will be very effective in defending us, so that we can be very happy here in Warangal. So thank you madam for taking us to the journey of how the animal is used as a model in research. Now it is a time for the Department of Zoology as a token of appreciation. We want to thank you. So I request all faculty members to come to the stage to say thanks to the madam.
బంద్ చేయమంటారు ఇంకా బ్యాక్గ్రౌండ్ స్క్రీన్ బంద్ చేస్తే మంచిగా ఒకవేళ తెల్ల తెల్ల రిఫ్లెక్ట్ అయిపోయినాయి అది దగ్గర రికార్డ్ కాదు క్లాప్స్ కొట్టండి మేడం సార్ చూడండి ఓకే సార్ ఓకే రెడీ సార్ ఓకే సార్ ఏంటిది మేడం
చెప్పరా
The next technical session, oral presentations are there. All of you load your presentations in the system here.
అయిపోయినాక పోతాం కదా అయిపోయినాక పోతాం కదా
Is there anyone who are going to present poster presentations today? No.
Okay, right.
So on behalf of this oral presentation, first oral presenter is 𝕃𝕀𝕍𝔼. Associate Professor of Department of Modeling, KGC. He is organizer of this oral session, and Professor 𝕃𝕀𝕍𝔼 will be the jury for this oral presentations.
ఈ తూకం ఏంటంటే
వచ్చిందా?
I'm an MTech Bioinformatics student from University of Hyderabad. I am here to present my work titled Identification and Validation of Targeting the C protein of Nipah virus under the supervision of Professor 𝕃𝕀𝕍𝔼. We will go with first introduction of the Nipah virus. It belongs to the Nipah virus genus of 𝕃𝕀𝕍𝔼 family. The members of the Nipah virus genus are Nipah virus and 𝕃𝕀𝕍𝔼 virus. Its genome consists of native single-stranded RNA of 18 kilobase by encoding 6 structure and 3 non-structural proteins. The 6 structural proteins are fusion proteins and glycoproteins, which are responsible for cellular attachment and post-cell entry. Matrix proteins mediate morphogenesis and budding. Nucleocapsid phosphoprotein and long polymerase helps in virus recall. Ribonucleic protein, which helps in viral replication and host entry. Now moving to the 3 non-structural proteins, which help E genes, in which E and W proteins are produced by mRNA editing, and C protein utilizes an alternative declared as the category C primary pathogen by WHO facilities up to 75%. Till date, no licensed antiviral or vaccine for humans is there for this particular.
Ms. Shah, so there is only 7 minutes for you, so go to objectives, better methods and coverage. Results and coverage. Introduction is 5 minutes. So important slides you focus.
The pathology and severity of this virus is the major clinical manifestation is severe hepatitis and acute respiratory distress syndrome. The path is followed through pathogenicity is that in case of acute respiratory distress syndrome, it follows through respiratory tract, then epithelial cells, then endothelial cells, then enter through blood vessels and various organs such as heart, kidney and spleen. And through second pathway that is olfactory nerve, it enters CNS and disrupts blood-brain barrier. Now my main target is C protein. As C protein is a non-structural accessory protein which consists of 166 amino acids. Its cellular localization is cytoplasmic and it is membrane association. The main functions and evidences for C protein is its modulates viral RNA synthesis via acting as a negative regulator of replication. It inhibits the interferon beta production in RIG1 MME signaling and interacts with TSG101 to promote virus budding. There have been studies reported in which C-deficient patients show reduced viral disease. As the regulatory accessory proteins are not targeted as a drug, but there are many examples in which these are targeted as drugs, such as NS5A inhibitors which disrupt replication complexes in HCV, NS1 inhibitors which modulate immune invasion in dengue, and EP35 inhibitors that block double-stranded RNA binding in Ebola. As no small molecule inhibitors reported, yet targeting C could be a block replication and immune evasion due to causes given below. Now this is the workflow that I had followed. First one is sequence retrieval and 3D model generation, followed by disorder characterization, binding pocket identification, virtual screening, then molecular dynamic simulation, trajectory analysis, enzyme evaluation, and drug identification. So here we perform sequential identity, the same genus 𝕃𝕀𝕍𝔼 virus and Nipah virus, and we found that there is 83% identity between them. So this conservation suggests that a single inhibitor can potentially target both the viruses for an inhibitor purposes. Now Now, as no structure was available in any databases for this, so we generated the 3D structure using AlphaFold 3 and validated it using CX program, and the Ramachandran plot showed a good result that no residues come into the disallowed regions. Now, we have done disorder prediction as this is the intrinsically disorder protein, and that means that the predicted disorder result involved in binding, and the baseline depicts that the predicted intrinsic disorder is a structural disorder. As disorder studies, we done this as it is a intrinsically disorder protein, and the most of the cases it functions while it interacts with host or any other substrates. Now we have done binding pocket identification. The various tools used are V2Hang, FTMap, and CASP-fold to predict druggable. And here are the binding residues that we got. Further, as we have seen that C protein is involved in viral replication, to check its helicase activity and keep nucleic acid docking, and we used HEDOC, which is a tool used for docking purposes of macromolecules, and we got that the same receptor ligand interface residues were there that we got through binding pocket prediction and all. Now we have chosen FDA approved library for our screening purpose, as in case of FDA approved library we don't have to do so much background work, and it is already most of the parameters are already designed for that, and it will be easy for the accelerated drug reprocessing strategy. And here we have done structured this virtual screening in which evaluation of the identification of favorable interaction profiles and prioritization based on time stability, and after virtual screening multiple parameter filtering framework was done. Now these are the interaction profile of that. We have an IVC protein and the compound. And the top 5 virtual screening results, and these were used for further molecular dynamic simulation. These are the stores and interacting residues. Now moving to molecular dynamic simulation, we use Schrödinger's rule for our molecular dynamics purpose, and we perform the 100 nanosecond simulation using a parasite and the stable temperature throughout the simulation. And here we obtain RMSD and RMSF stability, as we can see that the RMSD is gaining stability after the 60 nanosecond simulation between 0.4 to 0.6, which is the standard range for an intrinsically disordered protein. And in case of RMSD, we can show that after binding to ligand, it is showing stability, that the peak is lower than the echo form of protein. Now moving to hydrogen bonds, and our top is making an average of 2 to 14 hydrogen bonds, suggesting that it is stable and interacting throughout the simulation and not moving out of the pocket. And MMGBAC binary energy analysis has been done, and it also gives us strong negative binding energies, and calculation confirms strong negative thermodynamic binding energy for all optic. Further, we have done physicochemical properties, and these show a stable and acceptable reasons for all the parameters and all. Now, after that, admit and pharmacokinetic properties has been evaluated for top 3 compounds, and it's also showing the best, as we can see that in case of HERG that
last 1 minute 6
Now these are the conclusion. We predicted 3D structure of NIBC protein and validated this literature review functional resolution protein DNA docking. Structure this virtual screening identified the top hits, and the 100 nanosecond molecular simulation of the complexes with LIBC protein shows stability throughout the simulation time, and MMGBAC calculation shows strong binding energy for all 3 molecules, indicating that these inhibitors intact theory are stably with the targeted binding region. And from this result, we are further going through pharmacophore modeling, and we get the chemical that we need. So with the help of that, we can design the de novo drugs for this.
Thank you. Any questions on that?
Is it only simulation only in silico work, or have you done any practical work?
Only in silico.
Only in silico. Are you having any further plans?
Till now, I have only in silico plans, but in our lab, if we get the good result, it will be followed by experimental.
Thank you.
Good afternoon to everyone present here. I am 𝕃𝕀𝕍𝔼 from Biotechnology from School of Life Sciences, Department of Biotechnology and Bioinformatics under the supervisor Professor 𝕃𝕀𝕍𝔼. Present my project, Molecular and Biophysical Insights into Peptidase of 𝕼𝕍𝔼. To give an introduction, leishmaniasis is an intracellular parasite. Leishmaniasis is caused by an intracellular parasite that is transmitted to humans by the bite of a female 𝕼𝕍𝔼 species. It ranges in clinical manifestation from cutaneous to visceral leishmaniasis. Cutaneous and mucocutaneous are less severe, whereas the visceral leishmaniasis is fatal and highly lethal. According to the WHO report, cases occur in Brazil, East Africa and India, and estimated of about 50,000 to 90,000 of BL have been reported annually, and out of which only 25 to 45% have been reported individually to WHO. As per the WHO report of 2021, the leishmaniasis and HIV co-infection has been in a rise in Brazil, Ethiopia and Bihar. To give an introduction to the life cycle of Leishmania donovani, which is the species that leads or causes visceral leishmaniasis, it belongs to the order Nictinoplastida, family Trypanosomatida, genus Leishmania and species donovani. The sandfly, when it takes a blood meal from the infected individual, the non-motile amastigote form is taken up by the sandfly, and in the gut of the sandfly it is transformed into a motile promastigote form and reaches the proboscis. On reaching the proboscis, it is transmitted to a healthy individual when it goes to take a new blood meal from a non-infected individual. Like this, the life cycle once again continues between the two forms, that is the motile and the non-motile form. Talking about the existing treatments, that is the two major treatments, that is pentavalent antimonials and the liposomal amphotericin B are commonly used along with the combination therapy of parmomycin. But the biggest disadvantage is they cause infusion reactions as well as systemic toxicity and has to be monitored regularly with IM and IV injections. The most commonly used drug is metformin, but the relapse rate with metformin is really high, between 11 to 20%. So due to the substantial global burden, high mortality rate, drug toxicity and the emergence of drug-resistant leishmanial parasites, particularly against metformin, it highlights the need to characterize novel drug targets and identify novel molecules. In order to have novel targets, we have identified metallocarboxypeptidase from L-donovani, which belongs to the family of M32 carboxypeptidases. These carboxypeptidases remove the residues from the C-terminal end and releases the amino acids. These are essential for the nutrition of the parasite as well as for resistance against metformin. According to literature, it has been proved that these carboxypeptidases are only unique to lower eukaryotes, prokaryotes, and they are completely absent in higher organisms. So hence, it is only seen in green algae and trypanosomatics and leishmania. From the other part of literature, it has also been seen that the NCP of Trypanosoma brucei and cruzi have been targeted to inhibitors. We did a phylogenetic analysis between all the existing organisms that actually secrete metallocarboxypeptidases, and we have seen that Leishmania and Trypanosoma are almost belonging to sister clada. So hence making LDMCP a potential drug target to be studied against leishmaniasis. In order to study the metallocarboxypeptidase, we started with the cloning of LDMCP, that is a 1497 base pair, which was amplified from the genomic DNA of Leishmania donovani and with the flanking ends of ND1 and HIMB3 sites. It was cloned into PET21A expression vector and it was transformed into the DL21DE3 expression cell line. The positive clones were then used to express the protein with an N-terminal 6S histidine tag with a size of 56 kilodaltons, and it was induced at 0.1 millimolar IQTG at 18 degrees centigrade for 20 hours. After the expression, it was purified through nickel anti-affinity chromatography and it was optimized for homogeneity. The homogeneous protein and the LU fractions were dialyzed and then concentrated using a 30 kilodalton molecular weight cutoff centricon. These are the respective SDS page gels of the induction as well as the purification. The purified protein was then passed through a column, that is a size exclusion chromatography column, Superdex 16 by 600 200 Pg using FPLC, and the oligomeric state was analyzed by plotting the elution volume that was obtained approximately at around 79 ml across the standards. So when the molecular weight was determined, it was close to 112 kilodaltons. That was exactly double of the one obtained in denaturing gel electrophoresis. So hence we concluded that the protein exists as a homodimer in solution. After determining the oligomeric state, we went forward to characterize the secondary structure of the protein through circular dichroism. The intrinsic UV-CD was taken between 260 nanometers and 190 nanometer wavelength at a pH of 7.5, and we obtained a maximum percentage of alpha helices. This was corroborative and correlated with the predicted structure using SAI database. After this, the thermal denaturation was also performed using the same series circular dichroism spectroscopy, and the temperature was varied between 20 to 80 degrees centigrade, and the partial unfolding exactly of 50% of the protein, that is the melting temperature of the protein, was obtained at 49 degrees centigrade. The stability of the protein at varied pH was also checked through circular dichroism, and there was seen a considerable loss of alpha helices from 60% to 38% only in the acidic range towards 3.5. At the basic range and neutral range, the protein was highly stable. After the circular dichroism analysis, we went forward
And today I'll be presenting a comparative study of carbapenem-resistant genes in Klebsiella pneumoniae from Indian clinical isolates. Klebsiella pneumoniae, a gram-negative bacteria, is a major cause of hospital-acquired infections, especially in intensive care settings, and it causes major urinary tract diseases and bloodstream diseases and even eye infections. And carbapenems, which are broad-spectrum beta-lactam antibiotics, they are the last resort antibiotics to treat diseases, certain diseases like this caused by Klebsiella pneumoniae. But increasing carbapenem resistance of Klebsiella pneumoniae limits treatment options, because then we have to use combination treatments which might be complex, or use less effective and more toxic treatments, so that is not ideal. India in particular reports a high burden of carbapenem resistant isolates, and which is why genomic surveillance is critical to understand the resistance patterns and the spread patterns of major antibiotic resistance determinants in Klebsiella pneumoniae. As we have established that carbapenem resistance in India is widely reported, the data is scattered. The data that there there are carbapenem resistance genes in Klebsiella pneumoniae is there, but it is scattered, which is why systematic comparative screening of Indian genomic data sets is limited. The data that is available in websites and repositories such as NCBI, screening of that data remains limited. So repositories such as NCBI, they are underutilized for these kind of screenings. So there is a need for scalable and there is a need for automated bioinformatic analysis pipelines to understand these kind of studies. So the objective of this study includes retrieving the Indian clinical Klebsiella pneumoniae genome assemblies from NCBI and then screen for major carbapenemase genes using compare them across the different variants and isolates, and then develop an automated and reproducible screening workflow. A total of 64 publicly available Indian clinical isolates were taken from NCBI, downloaded from NCBI website, and the inclusion criteria includes that it must be from a clinical origin, so various urine samples, blood samples, and even sterile samples were taken from NCBI, and it must be of Indian region, and the genome assemblies must be complete or high quality draft assemblies. This criteria was mentioned and it is publicly on the NCBI website. And lastly, all of these files were downloaded in FASTA format to genome assemblies from NCBI, and then we scan them for resistance genes using bioinformatics tools such as Aggregate, which is a resistance gene scanning. The reference database for antimicrobial resistance genes in Aggregate is ResFinder. So the genomes that were retrieved from NCBI assemblies were compared with the ResFinder database in Aggregate, using Aggregate. The target genes that were extracted from this study from these genomes are 𝕃-Indian, 𝕃-OXA48-like, and 𝕃-KBC. Because it is epidemiologically and clinically reported that these are the major carbapenemase resistance genes in India. And finally, we do a frequency analysis to compare the cooperates. The pipeline looks something like this. So to ensure that there is reproducibility and scalability, batch processing was done of 64 genome assemblies, and a shell-based loop was created so that it automates screening all of the 64 genomes at a time. And then a strict detection threshold was applied of more than or equal to 90% identity and 90% coverage, so that the results or output have high confidence. The results look something like this. So out of the total 64 isolates, it was seen that 23 of them had 𝕃-Indian gene and 15 of them had 𝕃-OXA48-like gene. And none of them had 𝕃-KBC, which is aligns with the reports that are publicly available. When we look at the cooperative pattern, 16 of the 64 genome clinical isolates, they showed only NDM, while 8 of them showed OXA48-like. While there is an overlap that was seen between NDM and OXA48-like. Notably, 33 of the 64 isolates did not have any of the major carbapenem resistance determinants. So from the results, we can interpret that 𝕃-Indian is the most prevalent in India. It showed 36% of the data set had 𝕃-Indian isolate, and OXA48-like variants were identified in 23% of the isolates, while cooperates was seen in 7 isolates, which amounts to 11%. Notably, 52% of the isolates did not have any 2 of these or cooperates of these, which suggests that there might be other resistance mechanisms that might be developing, which is a major concern to be studied. And these findings and results are consistent they align with the reports that are publicly available about these isolates. So future work on this study can include larger Indian genomic collections and to perform co-occurrence and plasmid localization analysis, so we can find out if the genes are plasmid based or not, and then we can develop mechanisms to tackle it. And then to integrate phylogenetic clustering to study the transmission patterns. And lastly, to develop a scalable AMR genomic surveillance framework, because this is highly required for huge data sets. In summary, 𝕼𝕼 was the most prevalent 𝕃𝕀𝕍𝔼 that was detected among the Indian clinical isolates. OXA48-like variants were identified in a significant amount of isolates, and the overlap or co-occurrence of NDM and OXA48-like was observed in 11% of the isolates. And this shows that an automated bioinformatics screening analysis enables a proper genomic surveillance, which is important, especially when we talk about organisms like Klebsiella pneumoniae, which are developing resistance at a very fast rate. Thank you. It's an in silico analysis. Yes, that's right.
What stage you will follow?
Now, it's not supported by anybody. I'm doing this on my own. So, if I'm at a stage where it would be possible for me to work in a wet lab.
There is no guide for you there? You got your own?
Yes.
Is it a part of project work?
No. We have a subject called bioinformatics, so I was just interested in it. So I'm still learning about it. I was interested in this. Next.
Because of this modifications, it causes the variability in the drug concentration. In case of some, it might cause it, the concentration may be increased or decreased to less that the impact of the drug may not be possible for the patient. So it causes major clinical outcomes or the reduced efficiency or the adverse drug reactions. The comparative genomic analysis, how it can be treated is we use a bioinformatic tool to overcome this situation. First we do the sequence read and we do the quality control on it. Then we do the alignment of the genome sequences. We clean up the alignment and the variant column where the variations have been occurred. The genomic comparison of the genome variants across the individuals or the population is done with the genomic sequence. And we identify the clinically relevant pharmacogenomic markers. These markers represent the measurable DNA or the RNA characteristics which will tell how the gene is in contact with the drug and how it will affect. So based on this identification, we detect the population-specific genome signatures. On this, we can create a large-scale analysis of the bioinformatics content, and we can do the establishment of the genotype-phenotype correlation as well as the drug response, so that drug response, drug discovery can be done as a register basis. Pharmacogenomic data integration. When it comes to integration, the acquired genomic data, we compare it with the publicly available genomic databases, which give the information of the available information about in the public. The integration with the curated responses such as the 𝕃𝕀 and the 1000 gene product, it enables the prediction of the altered drug metabolism and the adverse drug reactions associated. Coming to this application and the future impact of this topic, mainly in case of the oncology, it helps to optimize the chemotherapy efficiency and reduces the toxicity so that it helps in further treatment in case of the oncology. Cardiovascular care, it guides on personalized anticoagulant therapy. In case of the infectious diseases, it predicts variability in antiviral drug response. In case of ADR prevention, it identifies high risk for genotypes to improve safety. Genotype guided therapy, it enables personalized drug selection and dosing. In precision medicine, the advanced selective and the individualization therapy. So in general, the integration of the bioinformatics tools in comparative, it helps to create personalized medicine for the user. Thank you.
That's good.
My name is Pranati. I'm studying third year Videography. Today my presentation is about comparative bioinformatic analysis of stress response genes in extremophilic microorganisms. So coming to the introduction, what are extremophils? These are the organisms that thrive in environments characterized by conditions. What are those extreme conditions? Those are high temperature, high salinity, high pressure and high pH. Take an example. First, thermos aquaticus organism can survive in high temperature because it has a enzyme, and whereas E. coli cannot survive in those conditions because they lack heat shock proteins. Classification of extremophiles based on the environmental conditions are thermophiles. They are adapted to high heat environments, Halophiles, they thrive in high salinity, salty conditions. Cyclophiles are adapted to extreme cold such as -5 to -20 degrees. Acidophiles, they are the organisms which live in low pH conditions. So how do we compare? Coming to the methodology, the first step is we select the data from major resources like NCBI or GenDrive. What is NCBI? NCBI is the National Center for Biotechnology Information. It contains whole genome sequences. So how do we select? We check for the complete genome availability. Then we check the quality of the proper annotation, and we then characterize the strains based on ecology. The second step is the identification of stress response genes. We identify these stress response genes using homology based screening, where no reference sequences are compared against selected genomes using sequences like 𝕊ℝ𝔼𝔼 or FASTA. In 𝕊ℝ𝔼𝔼 we use hits based on the different threshold parameters. Next, we check for the functional annotation. Predicted genes are validated through conserved domain analysis and functionally classified using gene ontology. What is gene ontology? Gene ontology is a classification based on characterization, mapping, protein coding sequences to specific stress-related biological processes. The next step, we compare the genomic analysis of the organisms in externophiles and the gene we compare with NCBI. We evaluate the gene distribution across extenophile groups, examining presence, absence patterns, copy number variations, and identify conserved versus specific genetic signatures. Then we construct phylogenetic trees using multiple sequence alignment. We enable this assessment of evolutionary relationship, diversity patterns, and environmental adaptation based clustering. What is the in brief? What is the methodology? Is first we select the extenophilic organisms, then we retrieve the data from NCBI and GenDrive, then we identify the stress response genes using BLASTA or FAST, then we do the functional annotation, like we check whether the copy number is matching or conserved domain is there or not, then we compare the genomic analysis of those extenophiles versus the normal, and then we construct the phylogenetic relationship. What are the evolutionary insights which we get after comparing is the clustering patterns based on environmental adaptation, evolutionary divergence in stress-specific gene families, conserved evolutionary lineage in core stress genes. Let us look at an example which is of the organism Cyclomonas ingrahami, which is a cyclophile. These cyclophiles live in extreme cold conditions such as -5 to -20 degrees. When we compare these genes with the normal genes from NCBI, what do we observe? We observe that the increased in Cyclomonas ingrahami, there are increased cold shock protein gene families, and genes regulating the membrane fluidity are also increased, and reduced proline content in proteins. What does this proline do? Proline improve the flexibility of protein folding, in which if the proline content is reduced, then the folding of protein stays intact. What are the key findings from this? The first is we find luminously conserved genes identified, such as molecular chaperones. Example, HSP family, which is rich on proteins, and GROEL. Next is DNA repair proteins, which are like A and UVR systems. The next finding is environment-specific genetic signatures. In halophiles, we find that there is enhanced protein folding machinery. In halophiles, osmoprotected biosynthesis genes. Cyclophiles are cold shock proteins. Acidophiles, proton effects and pH regulating genes. What are the applications of these comparisons? First, we use in industrial biotechnology, which is we use thermostable enzymes, as I mentioned earlier. Example is Thermus aquaticus. Then we use in bioremediation. In bioremediation, there is a tolerance in harsh environments. In pharmaceutical engineering, we use it for enzymes. And in astrobiology, we use models for extraterrestrial survival. Coming to the conclusion, Stress response genes are central to extremophilic survival. Core genes are evolutionarily conserved. Specific adaptation reflects the genomic plasticity. Bioinformatics provides powerful insight to molecular evolution. Carry on extraterrestrial environment. Myself Madhuri, I am third year biotechnology. Today my topic is about fish as living bioreactor, harnessing fish associated microorganisms for aquatic bioremediation. The main problem in our society now is aquatic pollution. The main reasons for the aquatic pollution are industrial discharge, which contaminates water bodies, and agricultural runoffs, which introduces nutrients and chemicals. Aquaculture waste increases ammonia toxicity. These pollutants threaten aquatic life and human health. There is the only solution for this aquatic pollution now is bioremediation. It uses microorganisms to remove the pollutant. It is eco-friendly and also sustainable approach. It converts the harmful components into the safer forms. Here we use fish as a bioreactor, a natural treatment system. Fish continuously interact with the surrounding water, which host beneficial microbial communities, self-sustaining biological systems. Here fish with the microbes are the continuous remediation unit. Fish associated microorganisms. The microorganisms which present on the skin, which comes into the contact in water, gill microbes, they expose it to dissolved substances. The gut microbes, they process organic matter. These pollutants are, they remove ammonia and organic waste and by degradation. They absorb the heavy metals and biodesorption and biofilm formation. Applications of this topic is mainly aquaculture water quality, and it reduces the disease outbreaks. It improves the fish growth. It reduces the antibiotic usage, and also the advantages are eco-friendly and cost-effective, and also it is a continuous treatment. Future scope is engineered microbiomes for pollutant removal and probiotic and biofloc based aquaculture. Heat metal removal using biosorption, restoration of water bodies, climate resilient aquaculture systems.
Thank you. Anybody?
There are 6 presenters. All the presenters are almost all second year nuclear PhD process only. They are not researchers, but they have research knowledge. They presented very well. All the presenters, I congratulate for this presentation. This stage, I thank organizer, Department of Zoology,
convener Hiroyunagar and today's judge and senior professor Shamita Madam Garu are judging the oral presentations. Thank you very much.
Oral presentations have been completed. So tomorrow again on day 3, the session starts at sharp 10. To be present by 9:40 in the afternoon.
మేడం ఇక్కడ చూడండి. రెడీ మేడం? లేదు వద్దట 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 క్లోజ్ చేయి మేడం వద్దనే వద్దాను వద్దండి క్లోజ్ చేస్తాం ఎవరు క్లోజ్ చేద్దాం వద్దంటాను మేడం.
Congratulations to all of you. You're all the way from Hyderabad.
इसको है पूरे तत्व कलर राइट साइड पे तो रोड पर स्माइलिंग मैडम सुना देखा कुमार అప్పుడు పంపిస్తారు కూడా అంతే అంతే మేడం అంతే వాట్సాప్ చేస్తే అయిపోతుంది ఫోటో తీసి వాట్సాప్ పంపితే బయట బెస్ట్ అయిపోతుంది దానికి ఇంత గోపే ప్లీజ్ గోపే నేనా కన్నా నమో శివాయ